Hello, my dear doctors. I hope you all are doing good. So, welcome here, my dear friends. This is Dr. Azam here, your faculty of anatomy and biochemistry. And today we are here to revise the entire biochemistry here. And you all know very well the students who are preparing for the January 2025 exam, 12 January 2025 exam. Biochemistry is one of the very important subject, and directly you can score. Like most of the students who have responded me in the previous exams and all, they're like, you'll be able to answer very quickly and save your time when you have to solve the other subjects questions and all. So, my dear friends, without wasting any further time, initially there was a little bit glitch joining there. I was trying it and now it is rectified. So, let's get started. Okay. So, first of all, to begin with biochemistry, you all know very well from biochemistry, you'll be approximately getting like 17 marks. But from last like couple of exams, they have been giving almost like 20 questions from there. And in this session, my dear friends, for the next couple of hours, okay, no, for next 2 3 hours, just be with me, sit back, relax, and enjoy the concept along with me because I am actually going to discuss like almost all the topics of biochemistry over here. So we are going to discuss about the electron transport chain, carbohydrate metabolism, protein, lipid metabolism, and the nucleotide metabolism. Followed by that, we are also going to discuss about the enzymes topic and the most important topic in biochemistry, that is vitamins. And from there are totally like 5 questions which have been asked in the last exam. Is it okay? So now, my dear friends, yes, thank you for your patience over there. Without any further delay, let's get started right now. Okay. So the first topic that we are going to discuss is electron transport chain. And my dear friends, remember from the electron transport chain, definitely you can expect like one question from here. Fine. Now, first point that you have to remember here is that electron transport chain, remember, it is also known as the respiratory chain. Sir. It's also known as the respiratory chain. Now, in the electron transport chain, where it is going to take place, everyone knows that it is taking place in the mitochondria. But remember that it is going to take place in the inner membrane of mitochondria. On the inner membrane of the mitochondria. That is important thing. After that, we all know the electron transport chain has got totally like 5 complexes. Complex number 1 is known as the NADH dehydrogenase. Complex number 2 will be succinate dehydrogenase. Complex number 3 will be cytochrome reductase. And the complex number 4 will be cytochrome oxidase. And the complex number 5 will be FOF1 particle. FOF1 particle. Fine. Now, now once you know the location and you know that there are like totally 5 complexes here, yes, the entire mechanism is not that much important. The important point to remember here, all these 5 complexes are made up of protein, and the complex with the copper is nothing but the complex number 4. Sir. The complex which consists of copper is complex number 4, already asked in your exam, sir. The complex which does not transport the H plus ions across the membrane, that will be the complex number 2, already asked in your exam. Complex number 2 will not transport the H plus ions. Please remember that one. Then after that, the final acceptor of electrons in the electron transport chain here, that will be the oxygen, my dear friends. Fine. Right on the complex number 4, what is going to happen? Oxygen is the one which is going to finally accept the hydrogen to form the water. Now remember, that is the reason why on the complex number 4, what is the reaction that is going to take place? Oxidation. And on the complex number 5, the reaction that is going to take place here will be the phosphorylation, because the phosphate group will be added over there. So therefore, oxidation and phosphorylation, both the reactions will take place together. That is the reason why the formation of ATP here, this process is known as the oxidative phosphorylation. Oxidative phosphorylation. Fine. So please remember the 3 MCQs here. The complex which consists of copper will be the complex number 4. And the complex which does not transport the H plus ions will be complex number 2. And then the final acceptor of the electrons over here will be the oxygen here. Now in the mechanism, I want you to remember these 3 points here for your FMG exam, guys. Done? Now once you are done with this one, now I will tell you the golden thing. In electron transport chain, one question will come for sure, my dear friends, and that question will be either from inhibitors of electron transport chain or from the uncoupler. So now get ready for one question in your exam in January here. Okay. Now the first thing here, complex number 1. Complex number 1 will be inhibited by phenobarbitone, and then Complex number 1 will be inhibited by phenobarbital and then rotenone. Phenobarbital, not one of the barbiturates. The barbiturates will be actually going to inhibit the complex number 1. And then rotenone. Rotenone is also going to inhibit the complex number 1. Please look at the spelling there. ONE1. Yes, that is going to inhibit the complex number 1. Along with that, remember amobarbital as well as the pyridine A. Then malonate is going to inhibit the complex number 2. And complex number 3 will be inhibited by British anti-levicide and the anti-mycin. British anti-levicide and anti-mycin. And then the most important one, the complex number 4 here, my dear friends. Complex number 4 will be inhibited by carbon monoxide, hydrogen sulfide and cyanide. I repeat again, carbon monoxide, hydrogen sulfide and then the cyanide. Already this question has been given repeatedly in your FMG exam. Please remember that. That is very, very much important here, sir. Fine. Now, after that, my dear friends, remember the complex number 5 is inhibited by oligomycin, and that is the reason why, remember, oligomycin, because of that reason only, complex number 5 is referred to as what? The complex number 5 will be referred to as your, yes, FOF1 particle. FO stands for oligomycin there, guys. I hope it is very, very clear, my dear friend. Okay. Now, now once you are done with all these inhibitors over here, all these inhibitors are done here. Remember, one question will definitely come from here, my dear friends. Definitely come from here. Is it okay? That's very, very important one. Apart from that, yes, uncoupler is also very much important. I told you that on the complex number 4, there'll be oxidation reaction. And on the complex number 5, there'll be phosphorylation. Oxidation and phosphorylation will occur together. They are like coupled reaction. Okay, coupled reaction. But in the middle of that, there'll be like uncoupler. Uncoupler is going to uncouple those oxidation as well as phosphorylation reaction, guys. Okay, so please remember the examples of that one. The artificial uncouplers will be the 2,4 dinitrophenol, 2,4 DNP, and even aspirin is also acting like an uncoupler in high doses. In high doses, my dear friend. And then even valinomycin and then nalinomycin as well as nijaricin, all these are the important uncouplers that you have to remember for your exam. Now, apart from this one, we also have physiological uncouplers, which will be actually present or produced in our body. Number 1, thermogenin. Already आपके FMG exam में पूछा गया है. And recently they also have asked about the location of this one. Remember, thermogenin will be located in the brown fat, the brown adipose tissue. Perfectly done. And thyroxine is also asked in your exam. T4. T4 is also acting like a uncoupler. And then the free fatty acid as well as bilirubin. So my dear doctors, please remember the examples of this one. Very, very much important. Is it okay? And by this, we have revised the first topic, that is electron transport chain. And yes, 1 mark for sure. Please revise inhibitors before going to the exam as well as the uncouplers. Inhibitors as well as uncouplers. Please remember this one here. Now moving ahead with one of the question which is asked in your exam recently in FMG June 2022, they have asked which of the following is a complex 4 inhibitor of electron transport chain. Yes, can you see there? Can you see the options and tell me the answer right now, sir? Complex number 4 will be inhibited by cyanide. Cyanide is going to inhibit the complex number 4. Perfect, perfect. Very nice. You're giving me the right answers over there. Okay, now after completing the electron transport chain, 1 mark has been secured right now, my dear friends. Okay, now let us go ahead with the next topic, that is carbohydrate metabolism. Let us discuss about the carbohydrates. Okay, तो इस तरीके से don't worry at all, just आराम से बैठिए और सुनते जाइए, because I am going to do the entire biochemistry like topic by topic, so it will be like very much easier for you to revise this before your exams and get set ready for like almost 20 marks in your exams. Okay, चलिए आगे बढ़ते हैं. Now, please look at this question here. First of all, a beautiful question, my dear friends. A middle-aged patient presented with high anion gap metabolic acidosis. Okay, there is metabolic acidosis here, and diagnosed with a defect of PDH complex. Which of the following dietary advice will you prescribe? Okay, now if you have attended my biochemistry class, my dear friends, in the carbohydrate metabolism, I have given you on the very first page, I have given you the overview there, guys. Now, what is going to happen? First, you'll be having glucose. Glucose will undergo certain reactions here, and finally, it is going to form what, sir? Pyruvate. That is nothing but glycolysis. And then this pyruvate will get converted to acetyl-CoA. That is a link reaction, only one single reaction. And here the name of the enzyme will be pyruvate dehydrogenase. Okay. And now this acetyl-CoA will actually enter into the Krebs cycle. ओके आपको याद होगा मैंने आपको ओवरव्यू में बताया था वो. तो ग्लाइकोलाइसिस and then the link reaction and then finally my dear friends remember the Krebs cycle. Now what is the question which is given here? There is a defect in the pyruvate dehydrogenase complex. Now if there is a defect here in the pyruvate dehydrogenase complex, yes my dear doctors, tell me here what is going to happen? The pyruvate level will actually increase. And once the pyruvate will increase, I told you that this is all going to take place in the presence of oxygen. Okay. But now, what is going to happen? If this is like excess over here, the pyruvate will now get converted to what? Yes, lactate. Lactate. And lactate, there is no further metabolism of that. Now, lactate is going to accumulate and that will lead to what? Acidosis. So, metabolic acidosis explains in this manner if there is a defect in the pyruvate dehydrogenase complex. Okay, acidosis यहाँ पर हो रहा है. तो अब आप मुझे बताइए like what is the diet that you'll be prescribing to this person? Now, only carbohydrate intake, option number one, no way, no way. We are not at all going to prescribe this one here. Okay. Then high carbohydrate, low fat intake, no carbohydrates. So these two options are eliminated. We have to give low carbs. Why? Because carbs are not getting, metabolized here. But now the question here is out of these two, high fat or the high protein, which one will be like more better? Which one will be more better option here in this one? Of course, there is low carbs. Yes, please remember that. Yes, high fat and the low carbohydrate diet, that one we have to actually prescribe here. So the overview that I have given you, my dear friends, from that overview, one question here, yes, done. Now, after that, moving ahead, yes, in carbohydrate metabolism, we have got like many pathways. The first thing that you have to learn in all these pathways over here is that, yes, my dear friends, the location of this one as well as the rate limiting enzyme. First, let us just quickly go through this one. Why? Because this almost many of the students will be knowing this one. Now, what is the another name of glycolysis? It is also known as Embden-Meyerhof pathway. Please remember that. We know glycolysis will take place in cytosol. And yes, what is the rate limiting enzyme? The very famous one, PFK1, phosphofructokinase 1. Okay. And after that, the second one is the Krebs cycle. Krebs cycle is also known as the tricarboxylic acid cycle, also known as the citric acid cycle. Where is the Krebs cycle taking place? Mitochondrial matrix. Yes. What is the rate limiting enzyme of this one? That is isocitrate dehydrogenase will be the rate limiting enzyme, IDH. Okay. The next one, gluconeogenesis, is the one where we are going to form the glucose from the non-carbohydrate sources. Non-carbohydrate sources. And please remember those non-carbohydrate sources with the mnemonic as LPGA. So we can form the glucose from the lactic acid, from the pyruvic acid, and from the glycerol. And then finally, A stands for all the amino acids can form the glucose, except two amino acid. One will be the leucine, another one will be the lysine. Except these two, all of them can form the glucose. Why? Because leucine and lysine will be the, yes, ketogenic amino acids, my dear friend. Gluconeogenesis will take place both in liver as well as kidney. Majority 90% will be in the liver, 10% will be in the kidney, both in cytoplasm as well as mitochondria. I will show you the reactions within few minutes, and along with that, we are also going to see the clinical correlation also, my dear friends. And yes, what is the rate limiting enzyme of gluconeogenesis? Fructose 1,6 bisphosphatase will be the rate limiting enzyme. Fructose 1,6 bisphosphatase. Now, after that, glycogenesis, glycogenolysis, formation of glycogen, breakdown of glycogen. यार मुझे ना ये बताओ, we have been learning from school days that glycogen is the storage form of the glucose. Where it will be stored? So remember, it will be stored in number one, liver as well as the skeletal muscle. So from here, apply common sense. Of course, if it is getting stored there, it will be forming there and it will be even breaking down there only. So glycogenesis will take place in liver and skeletal muscle. Glycogenolysis will be taking place in liver and skeletal muscles. Fine. Now, the rate limiting enzyme is very much easier over here. Glycogenesis, yes, we are forming glycogen. So what is the rate limiting enzyme here? Glycogen synthase. Glycogen synthase here, sir. ठीक है ना? Now, after that, my dear friends, glycogenolysis, what is the rate limiting enzyme? Glycogen phosphorylase is the rate limiting enzyme. Glycogen phosphorylase is the rate limiting enzyme here, sir. Then HMP shunt, very much important one, which is also known as pentose phosphate pathway. Of course, it is going to take place in the cytoplasm. Please remember this one here, already asked in your exam. And the very famous enzyme G6PD will be present in this pathway only, and that is the rate limiting enzyme. So what you have to do here is, yes, my dear friends, this tabular form, you have to revise that so that you'll be able to answer the questions based on location as well as the rate limiting enzymes. Okay. So you are able to, yes, revise the locations here as well as the rate limiting enzyme. This is important here, sir. Done. Now, once you are done with this one, let us discuss about all of them in detail along with the clinical correlations also, guys, one by one. ठीक है ना? चलिए आगे बढ़ते हैं. Now, in this diagram, I am showing you on one side glycolysis, conversion of glucose to pyruvate. On the other side, gluconeogenesis. Gluconeogenesis. Okay. Now, my dear friends, remember in glycolysis, I'll tell you like 2-3 points which are very much important here. The first thing is already we have studied in glycolysis what is the rate limiting enzyme. It is PFK1, phosphofructokinase 1. ठीक है ना? And then number two, what is the net gain of ATP? So please remember, if it is aerobic glycolysis, there will be totally 7 ATPs will be obtained here. And if it is anaerobic glycolysis, there will be net gain of 2 ATPs, my dear friends. That is important here. Then do you have any inhibitor of glycolysis? Yes, the inhibitor will be the fluoride. And fluoride is going to inhibit which enzyme of glycolysis? That is enolase. फ्लोराइड इज गोइंग टू इनहिबिट इनोलेस. लेकिन आपके एग्जाम में सवाल कैसे दिया जाता है? NaF, सोडियम फ्लोराइड. तो सोडियम फ्लोराइड इज गोइंग टू इनहिबिट विच एंजाइम? इनोलेस. दैट इज एन इंपॉर्टेंट पॉइंट टू रिमेंबर, माय डियर फ्रेंड्स. Okay, now once you are done with this one here, now let's have a comparison with the gluconeogenesis where we are going to convert the pyruvate back to the glucose here, sir. And we all know the next one. How many irreversible reactions do you have here in the glycolysis? Remember, there are totally 3 irreversible reactions. What are the 3 irreversible reactions? The first one, HKGK, hexokinase and the glucokinase. And then the phosphofructokinase 1, PFK1, that is a rate limiting enzyme. And then finally the PK, the last one, pyruvate kinase. So hexokinase, glucokinase, as well as the PFK1, as well as, remember, the pyruvate kinase. These 3 will be the irreversible reaction. Now, if these 3 reactions are irreversible reaction, अगर ये तीनों irreversible reaction है तो क्या किया जाएगा? In gluconeogenesis, we need to learn how to reverse them. So number one, pyruvate is converted back to PEP by an enzyme that is important here, that is pyruvate carboxylase. Very much important to remember. Why? Because this pyruvate carboxylase requires biotin as a coenzyme. Vitamin B7, biotin is acting like a coenzyme. So in the entire biochemistry, we have to remember like 3 enzymes which will be utilizing biotin, and biotin is required for which one here, sir? Carboxylases, guys. Carboxylases, fine. Then number 2, you have to remember, yes, fructose 1,6 bisphosphate. मैंने आपको अभी बताया था, this is the rate limiting enzyme of gluconeogenesis. And number 3, you have to remember about conversion of glucose 6-phosphate to glucose is by glucose 6-phosphatase. And this one here is important. Why? Because it will be absent in skeletal muscles. It is absent in skeletal muscle here. Fine. So this one here will be absent in the skeletal muscle. That is the reason why what is going to happen. You will not be able to see gluconeogenesis taking place. Yes. My dear friends, gluconeogenesis will not be able to take place in the skeletal muscle. Why? Because, my dear friends, remember that this enzyme, glucose 6-phosphatase enzyme, will be absent. Are you understanding, my dear friends? Yeah. So first thing, I want you to remember like 3 MCQs here. MCQ number 1, biotin is required by which enzyme? Pyruvate carboxylase. Number 2, which is a rate limiting enzyme over here? That one is fructose 1,6 bisphosphatase. And number 3, which enzyme will be absent in the skeletal muscle? That enzyme will be the glucose 6-phosphatase enzyme. Glucose 6-phosphatase enzyme. Perfectly done. So in this manner, in a very short time, in one slide itself, we are able to learn the important points of glycolysis as well as gluconeogenesis. Glycolysis as well as gluconeogenesis. In this manner, I'm going to revise the entire thing in a simple manner over here. Fine. Next one. So once you are done with this one here, yes, the enzyme that is released in hypothalamus is, see, dekho, this is like a bouncer question here. I mean to say you don't have to learn about this one here in detail and all. Just remember the enzyme, like which type of this hexokinase will be released from hypothalamus, that is hexokinase 4 here. So now in your FMG exam, they are asking you, okay, what is the differences between the glucokinase here and the hexokinase here, the very first reaction, glucokinase and the hexokinase. So remember, my dear friends, glucokinase will be present only and only in the liver, as well as it is also present in the beta cells of pancreas. Fine. Whereas hexokinase will be present in all the tissues present in our body. Fine. Now the question that is asked in your exam is, what about the insulin here, my dear friend? Okay, insulin here. Insulin will be acting out of these 2 enzymes. Remember, insulin will be acting on, or it is going to induce which enzyme? Glucokinase को ये induce करता है. It is going to act only on the glucokinase, not on the hexokinase here. So glucokinase present in the liver. Yes, of course, insulin will be acting over there, my dear friends. So please remember, yes, insulin induces which enzyme here? It is going to induce the glucokinase. So this question is also covered over here, sir. That's all. Okay. By this, we have covered all the points regarding this one. Now, once you are done with the glycolysis as well as gluconeogenesis that I have shown you in the table there, my dear friends. Now, once you are done with this one, now let's get started with the glycogenesis and glycogenolysis. Formation of glycogen and the breakdown of glycogen here, sir. Now, we don't have to worry about the definition, you know definition very well. Glycogenesis means what? Formation of glycogen from glucose. We are going to form the glycogen from glucose. Glycogenolysis means what here, sir? That is nothing but breakdown of glycogen to glucose. Fine. Okay. And where is this taking place? I told you very clearly. Glycogen store कहाँ पर होता है? Liver और skeletal muscle में होता है. And both these pathways are taking place in the cytoplasm only. Cytoplasm only. Fine. Now let us quickly revise this one. That is the reason why you can see here in our LMR session here in this biochemistry last minute revision session here. I have written like maximum things on the slide over here. Why? Because my dear friends, we are not going to waste the time. Let us cover as much syllabus as possible here. And my students should be getting almost like all the questions from this video itself in the couple of hours. Yes, entire biochemistry will be revised. So that is why I have written everything ready-made here. So please observe very quickly here, sir. Now, in glycogenesis, what is going to happen? First, let us get started with the glucose here. Glucose will be activated to UDP glucose. And using that, we are going to form the linear chain of the glucose molecule. And that linear chain is formed by glycogen synthase enzyme. Very much important one. And after that, we know very well, glycogen is not a linear polymer. It is a branch polymer. So therefore, it is done by branching enzyme. And therefore, finally, it results in the formation of glycogen. Only 3 points to be remembered, my dear friend. Number one, सबसे पहले क्या करो अब यहाँ पर ग्लूकोज को एक्टिवेट किया जाएगा और नंबर 2 इसे लीनियर चेन फॉर्म किया जाएगा और फिर इससे क्या होगा ब्रांच यहाँ पर फॉर्म किया जाएगा. ओके, दैट्स ऑल. इंग्लिश में भी हो गया है, हिंदी में भी हो गया है. ठीक है ना? Fine here. So first activation done, then the linear chain formation, then the branching is done, my dear friends. And what is the rate limiting enzyme? Already we have discussed, that is glycogen synthase. When you're going to form the glycogen, of course it is glycogen synthase. Next one. Now after that, my dear friend, degradation of glycogen. Glycogen will first break down to limit dextrin with the help of an enzyme known as glycogen phosphorylase enzyme. Now in this, what is going to happen in limit dextrin? Glycogen phosphorylase is a very, very important enzyme, very good enzyme. It is going to remove all the glucose molecules in the linear chain, but it doesn't have the ability to remove the branch. So therefore, it is not able to remove the branch from there. Okay, understanding, my dear friend? So from the limit dextrin, the branch will not be removed there. So what will be the next thing? The next thing will be the debranching enzyme. So debranching enzyme is going to remove the branch from there. And after removing the branch, we are obviously going to get linear chain. And that linear chain is again broken down by the same enzyme glycogen phosphorylase here. And it is going to become glucose 1-phosphate. Now glucose 1-phosphate to glucose 6-phosphate, we are moving the phosphate group from 1 carbon to the 6 carbon on the same molecule glucose. The name of the enzyme will be what here? Mutase. And finally, my dear friend, removing that phosphate group, the name of the enzyme will be glucose 6-phosphatase. Glucose 6-phosphatase. So we have like quickly revised this one here, sir. Now, once you are done with the formation of glycogen and the breakdown of glycogen, now let us learn the most important one, that is glycogen storage diseases. Glycogen storage diseases. Okay. Now, my dear doctors, if there is a deficiency of this enzyme, if this enzyme is defective, glucose 6-phosphatase enzyme is defective, then it will lead to type 1 glycogen storage disease, that is your von Gig's disease. Von Gierke's disease, that is type 1. If there is a deficiency or defect in debranching enzyme, it will lead to type 3 glycogen storage disease. Type 3 will be the Cory's disease, my dear friend. Type 3 will be the Cory's disease. And then if there is a defect in the glycogen phosphorylase enzyme, then it will lead to type 6 glycogen storage disease, that is Her's disease. But remember, in Her's disease, there will be hepatic glycogen phosphorylase deficiency. Then type 5 in McCall disease, again the same enzyme will be deficient glycogen phosphorylase, but please remember that it will be the muscle glycogen phosphorylase. Muscle glycogen phosphorylase. Okay. Now, my dear friend, remember one more thing. If the branching enzyme is defective here, then what is going to happen? It will lead to type 4 glycogen storage disease, that is Anderson's disease will occur. So therefore, what is going to happen? If branching enzyme is deficient, only linear chains will get accumulated. Okay, that means in type 4, what is going to happen? In type 4, what is going to happen? There'll be accumulation of abnormal glycogen. It is not normal glycogen, abnormal glycogen without any branches. Only the linear chains will be actually accumulated there. Okay, चलिए अब हम आगे बढ़ेंगे. The only and only one which is left out right now is the type 2 one. Type 2 one. Okay. Now I told you glycogenesis will take place in cytoplasm. Glycogenolysis will also take place in cytoplasm. So basically, where are we right now? We are in the cytoplasm. Now in the cytoplasm, there is one of the cell organelle that will be the suicidal bag of the cell. Suicidal bag of the cell, that is nothing but your lysosome. Now, why I'm telling you about the lysosome? Why? Because a little bit of glycogen will also enter here. Hardly 1 to 2% of glycogen will enter here. And this glycogen will break down here in the presence of an enzyme. The name of the enzyme here will be acid maltase. And if this acid maltase is defective, it will lead to type 2 glycogen storage disease, that is Pompey's disease. That is Pompey's disease. Okay. I hope this is like very much clear here, my dear friends. So in this manner, that is the reason why keep on following my LMR session here. Yes, in one image, I'm trying to finish like many concepts over here. So therefore, my dear friends, now look at all the glycogen storage diseases. Type 1 will be the von Gierke's disease, that is due to deficiency of glucose 6-phosphatase. Type 2 will be the Pompey's disease, that is due to deficiency of acid maltase, which is also known as lysosomal glucosidase. Type 3 will be the Cory's disease. In Cory's disease, what is going to happen here in type 3? What is getting accumulated here? Limit dextrin is getting accumulated here. So therefore, it is known as what? Limit dextrinosis. Similarly, in type 4, the name of the disease is Anderson's disease. And which enzyme will be deficient? Branching enzyme will be deficient. Type 5, MECARD, muscle glycogen. And then type 6, HERS, it is hepatic glycogen phosphorylase enzyme is deficient. Now, how to remember this one? What you do is you try to learn these two together. That is like, M for M and that is H for H, my dear friend. So remember, in HERS disease, it is due to deficiency of hepatic glycogen phosphorylase, and MECARD disease, it is due to the defect of muscle glycogen phosphorylase. Then you learn these two together, yes, with the mnemonic as A, B, C, D. Anderson branching enzyme, and Cory's there will be debranching enzyme. Easy to remember this one here. And type 1, everyone knows that it's very famous one, glucose 6-phosphatase enzyme is deficient. And type 2, Pompey's, yes, accumulation of glycogen in the lysosome will take place here, sir. Perfectly done. Now, out of all this, remember, for your FMGE exam in January 2025, this one is very much important. Type 1 glycogen storage disease, that is von Gierke's disease. Now, let us discuss about that one in detail. So please look at this question here. Which of the following enzyme is found to be deficient in the von Gierke's disease? That is glucose 6-phosphatase. Fine. But sir, what are the clinical features of this one? Let us discuss the clinical features in detail. Now, my dear friends, common sense, if that enzyme is deficient, glycogen is unable to break down. So glycogen will be getting accumulated, my dear friend, in the liver. So what is going to happen to the size of the liver? Number one, there will be increase in size of the liver. Hepatomegaly will occur. And even remember here in von Gierke's disease, kidney is also enlarged. Number one. Number two, second thing here is if glycogen is not able to break down to glucose, yes, if glycogen is not able to break down to glucose, what is going to happen? The level of glucose will decrease, so it will be hypoglycemia. But please remember here, there will be severe hypoglycemia will take place here. There will be severe hypoglycemia will occur here. Why? Because the enzyme which is deficient here, glucose 6-phosphatase, is the same enzyme for glycogenolysis also and same enzyme for gluconeogenesis. So neither glycogenolysis is taking place, neither the glycogen is actually going to break down, and neither, my dear friend, remember, gluconeogenesis is taking place. No formation of glucose from the non-carbohydrate sources also. So therefore, my dear doctors, there will be severe hypoglycemia, very much important. Clear? The next one. I told you very clearly in this von Gierke's disease, what is the substance that is getting accumulated? That is glucose 6-phosphatase, phosphate will get accumulated. Now, for example, if this enzyme is deficient here, glucose 6-phosphatase, of course, this substance will be getting accumulated. Glucose 6-phosphate is getting accumulated. Now that glucose 6-phosphate which is accumulated over there, my dear friend, remember that will be diverted to some other pathways. Now which pathways it will be diverted? Number one, This glucose 6 phosphate will now undergo glycolysis. And once it is going to undergo glycolysis, the end product of glycolysis will be what, yes, sir? That is pyruvate. The end product of glycolysis will be the pyruvate here, sir. But now we have excess of this one. So therefore, there are multiple fates of pyruvate. Number one, pyruvate can get converted to lactate. That will lead to what? Lactic acidosis. Number two, pyruvate can get converted to acetyl-CoA by link reaction. And you know very well, acetyl-CoA is the starting substance for fatty acid synthesis. And acetyl-CoA is also the starting substance for your, yes, cholesterol synthesis here, my dear friend. So what is going to happen right now? Now let us come to the conclusions right now, my dear friends. Conclusion number one here, if lactate is getting accumulated, what will happen? It will lead to lactic acidosis. Wow. Lactic acidosis will occur in the type 1 glycogen storage disease. Number two, fatty acid synthesis and cholesterol. What is going to happen? There will be increased level of fats. Hyperlipidemia will occur. Hyperlipidemia will occur. है ना? Along with that, remember, yes, it will also lead to what? Hypercholesterolemia also. Hypercholesterolemia. So cholesterol level as well as the lipid level will increase. Because of that, what will happen? The faces will be appearing like rounded there. So therefore, this is referred to as doll-like faces. Yes, doll-like faces will be seen in the type 1 glycogen storage disease. Makes sense. Acha, when you have like more of fatty acid, more of the fatty acids there, that fatty acids will actually break down to form the end product, that is ketone bodies. And that is the reason why even the ketone bodies level will also increase, leading to ketosis. Ketosis भी यहाँ पर होता है sir. So in this manner, if you just follow this one here, my dear friend, the clinical features, all this will become easy. So what is happening in the type 1 glycogen storage disease here? Remember, lactic acidosis, hyperlipidemia, hypercholesterolemia, nothing but doll-like faces, as well as it will also lead to ketosis, my dear friend. Okay. And apart from that, even the uric acid level will also increase here. Remember, it will also lead to hyperuricemia will also occur. So in this manner, don't worry, my dear friends, someone was telling me there, okay, to discuss about the clinical correlation, like we are going to discuss like many clinical correlations, my dear friends, here. In each and every topic, wherever the clinical conditions are there, we are going to discuss. Of course, they are asking, yes, a lot of questions regarding this one here. Okay, chaliye. अब हम बढ़ते हैं आगे. So this is like very important one. Please remember all these features over here. Now after that, now welcome my dear friends here. Now whatever table that you're able to see here on the screen, from this table, I'm assuring you that one question will definitely come from here. So 1 mark for sure from this table here. Now in every exam, they're asking one or the other question about the galactose metabolism as well as about the fructose metabolism. Galactose and fructose. And last exam, if you remember in the July exam, there was a question about the galactosemia. I'm going to discuss everything in detail here, my dear friends. And I have given you a beautiful table here. बाद में इसको आप आराम से देख लेना just before going to the exam. Please remember this one here. Is it okay? Now, now first of all, what I will do is I'll just give you a simple glance about the galactose metabolism first. Fine. Galactose metabolism. Now in galactose metabolism, whenever a person is going to drink milk or milk products, of course, we know that in the milk, the moment the examiner is telling you about the milk or milk product, in the milk you'll be having a substance known as lactose. Lactose will get digested to form a molecule of glucose and a molecule of galactose. Now what is going to happen? This galactose will be absorbed inside our body. And once this galactose is absorbed inside our body after digestion, of course, your digestion कहाँ पर हो रहा है? So remember, this digestion will be actually taking place in the intestines, my dear friend. So once the digestion is done, that galactose will be absorbed. Now what is going to happen? That galactose metabolism will be taking place in two cells in our body. Number one, it will be taking place in the liver cells, and number two in the kidney. Of course, in the kidney, it will be taking place in the PCT, proximal convoluted tubules. Fine. Now, once the galactose will enter inside the cell, the first thing what we have to do here, sir, we need to trap the galactose. And to trap that, we are going to attach the phosphate group. Galactose 1-phosphate. And whenever you're going to add the phosphate group here, the name of the enzyme here, kinase, and that one will be galactokinase. तो जैसे ही galactose वहां पर enter होगा, आप उसको phosphate group लगा दोगे. Fine. And once you attach the phosphate group, my dear friend, that galactose will be actually trapped inside the cell. वहां पर trap हो जाता है. ठीक है ना? Now what we are going to do is, we are going to react this galactose here with UDP glucose. And once you are going to react this one, only what is going to happen, transfer is taking place here. The glucose will go in the place of galactose here, and galactose will come in the place of glucose here. Transfer is taking place here. So therefore what is going to happen, it will become glucose 1 phosphate here, and this is going to become UDP galactose here. And no need to study about the further things. Why? Because glucose 1 phosphate and all this will undergo some other metabolism. But the most important enzyme here, the name of the enzyme here will be the galput, that is galactose 1 phosphate uridyl transferase. Galactose 1 phosphate uridyl transferase. Okay, galactose 1 phosphate uridyl transferase. Done. Now, my dear friends, remember, if suppose this enzyme is deficient, अब हम देखेंगे sir. Once you have seen the entire metabolism over here, now let us see the defects here. If suppose galactokinase enzyme is deficient here, sir, if that is defective there, then what is going to happen? We are unable to trap the galactose. Okay. Now, if they are unable to trap the galactose, that galactose will be going out into the blood. Oh my God, that will lead to what here? Galactosemia. Increased level of galactose in the blood. That will lead to what? Galactosemia. फाइन. सो देयरफॉर माय डियर फ्रेंड, वेलकम हियर. यस, अब आराम से टेबल देखिए सर, क्योंकि एक एक मार्क बहुत इंपॉर्टेंट है. वन मार्क इज वेरी वेरी मच इंपॉर्टेंट हियर सर. ठीक है ना? तो प्लीज रिमेंबर माय डियर फ्रेंड, गैलेक्टोसीमिया में क्या होता है? विच एंजाइम विल बी डिफेक्टिव देयर? एंजाइम डेफिशिएंट इज गैलेक्टोकाइनेस. When galactokinase will be deficient there, then what is going to happen? Increased level of galactose in the blood. Okay, and that galactose will start going in the urine also. Increased level of galactose in the urine. And how you're going to test that? Benedict test will be positive for this one here. Now the most important thing, when the galactose level is increasing in the blood, that will start going to your eyes, and in the eyes it will lead to what? Oil drop cataract, my dear friend. I want every student to please listen here 200% here. There are like so many questions coming from this topic only, guys, from this point only. Okay, now in the lens what is going to happen? Once it is going into the lens here, in the lens what is going to happen? The galactose which goes into the lens, that will now be getting converted to what? Alcohol, and that alcohol will be galactitol. Now this galactitol, remember, it is also known as dulcitol. Now, how is this galactose converted to galactitol, sir? How is this converted to this alcohol? So please remember, galactose is actually aldehyde, and now that is getting converted to this alcohol, galactitol. How? Of course, aldehyde will be reduced to form what, sir? Galactitol there. So therefore, the name of the enzyme here will be the aldose reductase. So what is the name of the enzyme here? Aldose reductase. That enzyme will be present in the lens over here, converting the galactose to galactitol. Very, very much important, my dear friend. So already this enzyme name has been asked in your exam. Number one question. Number two, oil drop cataract will occur due to what here? Yes, remember, there will be no problem with galactose, my dear friend. Please don't mark the answer as galactose is leading to oil drop cataract. No, 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 no. Please remember, galactose will freely enter into the lens and freely come out of the lens. Nothing problem with the galactose. But if it is acted upon by that enzyme aldose reductase, it is getting converted to galactitol, that alcohol, and that alcohol is getting accumulated in the lens, leading to that cataract. So remember, oil drop cataract will occur due to which substance over here? It is due to the galactitol, which is also known as the dulcitol, my dear friend. Clear? So this is very much clear here. So in the exam, if they are giving you that Benedict test is positive in the urine there, galactose level is increased in the blood, and oil drop cataract, yes, my dear friend, the answer is galactosemia. Enzyme deficient is galactokinase. Now what is happening? now what is happening, my dear friends, if this galput is defective here? अगर मान लो कि galput is defective here, what is going to happen? If galput is defective here, ये reaction नहीं होगा. If this reaction will not occur here, galactose 1 phosphate level will increase. And this is the product. And if the product level increases here, by negative feedback mechanism, it is going to inhibit galactokinase. If that galactokinase again inhibited, what is going to happen here, my dear friends? Remember, if that is getting inhibited, again, we are not able to add the phosphate group. Again, the galactose will be going out into the blood. Okay, so therefore, my dear friend, again, what is going to happen? Increased level of galactose in the blood, increased level of galactose in the urine. Benedict test will be positive here. Oil drop cataract. यहाँ तक तो बिल्कुल same है. So now if you see the classical galactosemia and galactosemia, till here all the features are absolutely same. So sir, how to do the differential diagnosis? Yes, my dear friends, welcome to my class over here. I told you in this one, in this classical galactosemia, In classical galactosemia, what is getting accumulated here? Galactose 1-phosphate is getting accumulated. It's not galactose. Galactose 1-phosphate is getting accumulated, sir. So because this galactose 1-phosphate is getting accumulated, my dear friends, please concentrate on the phosphate. Phosphate is trapped there. If it is trapped, what is going to happen in the blood? In the blood, the phosphate level is going to decrease. So therefore, there will be hypophosphatemia. Now, if the hypophosphatemia will occur, अभी मैंने आपको बताया था, where is the metabolism of galactose taking place? In the liver as well as in the kidney there. So in the liver, what is going to happen there? If the liver cells are not working, decrease ATP, what is going to happen? Hepatomegaly and jaundice will occur. Hepatomegaly and jaundice will occur. And in the kidney, what is going to happen if the PCT cells are not working there? So therefore, what is going to happen? From the PCT cells, you are not able to reabsorb the substances over there. Reabsorption will not occur. So all the substances will be going out in the urine. Bicarbonate is going in the urine. Everything is going in the urine there. We are not able to reabsorb that one. And that is nothing but the Fanconi syndrome here. So Fanconi syndrome will also occur. But along with that, please remember, even hypoglycemia will also occur here. Glucose level will also go down. So now, if you look at my table over here, easily you can differentiate here between galactosemia and classical galactosemia. All these features are absolutely same till here. Yes, if these things are given here, yes, my dear friend, that is classical galactosemia. Enzyme deficient will be the GalPut. Galactose 6-phosphate uridyl transferase. Clear? Now, similarly here, let's see about the fructose metabolism right now. Now, in the fructose metabolism, again the same thing. Once the fructose will enter into the liver cells, my dear friend. Okay, now, into the liver cells. Or else, let me just show you once over here. Again the same thing. Whenever a person is going to consume like honey or like fruit juices, like sweet substances. So what do you have in the honey and fruit juices and all? Yes, of course, you'll be having the sucrose. And this sucrose will go into your stomach and intestine, getting digested to one molecule of glucose and one molecule of fructose. अब यार ये fructose का क्या होगा? Fructose will come inside our body. Now this fructose will enter into which cells? Please remember this one here. The only and only cells where the fructose metabolism will take place is in the liver cells. Only in the liver. That is very much important. It will help you a lot later on. So please remember that carefully. Now in the liver cells, what is going to happen? First, again, the same thing. We need to trap. And to trap, we have to add the phosphate group. So it is going to become like fructose 1-phosphate. And whenever the phosphate group is added, enzyme name will be kinase. It is fructokinase. Wow. So that is the reason why please learn conceptually, my dear friend. And after that, my dear friends, what is going to happen here? Fructose will actually going to split here. And it is going to split in the presence of an enzyme known as aldolase B. Aldolase B. That is very, very important enzyme. Aldolase B, my dear friends. Okay. Now that is going to split into again, it is going to split into DHAP, dihydroxyacetone phosphate, and then glyceraldehyde. But that is actually not required for us. Why? Because that will enter into some other metabolism. Your focus only should be on these two enzymes here, fructokinase as well as the aldolase B. Fructokinase and aldolase B here. Fine. Now again the same thing. Apply the same concept that I taught you just now. Agar fructokinase ka deficiency hoga, if that is deficient, obviously we are not able to add the phosphate group. If we are not adding the phosphate group, my dear friend, we are not able to trap this one here. We are unable to trap. Again, fructose will go out into the blood and fructose level will increase in the blood. And because the renal threshold of fructose is very less, it will quickly start, yes, excreting out into the urine. And that condition is known as fructose urea, essential fructose urea. Okay, now essential fructose urea, my dear friends. Fine. And if aldolase B is deficient, then fructose metabolism will not occur. That will lead to what here? Hereditary fructose intolerance. So first remember, essential fructose urea is due to deficiency of fructokinase, and hereditary fructose intolerance will occur due to defect in the aldolase B, my dear friends. Please remember that one. Take it up. Then after that, my dear friends, remember, again the same thing. What is going to happen in essential fructose urea? Fructose is increased in the blood. Fructose is going out in the urine. And therefore, which test will be positive there? Benedict test will be positive there. That is the only thing. That is the only thing. That is the reason why most of the time it will be actually going unnoticed. Unnoticed. That is why it is known as essential fructose urea. Okay, essential fructose urea कहते हैं, ठीक है ना? And after that, my dear friends, remember, the fructose is increasing in the blood here, fructose is going in the urine. That is absolutely same. But how to do the differential diagnosis here? Again, the same thing. What is getting accumulated? Fructose 1 phosphate is getting accumulated. And when fructose 1 phosphate is getting accumulated, again the same thing, phosphate group will be actually trapped. If phosphate group is trapped, leading to hypophosphatemia, there is decreased level of phosphate group in the blood, as well as remember, if that phosphate is less there, yes, my dear friend, remember fructose metabolism kahan par hota hai? Only and only in the liver. So therefore, you'll be able to see only hepatomegaly and jaundice here, but there will be no Fanconi syndrome. There will be no Fanconi syndrome over there. And then along with that, it will also lead to hypoglycemia. Are you understanding, my dear friends? That's the reason why I have given you a beautiful table over here. Yes, in this one table, you can easily do the differential diagnosis here, sir. You can easily do the differential diagnosis. And yes, 1 mark will surely come from here, my dear friend. 1 mark will surely come here, my dear friend, from this table. I'm telling you. Yes, I think now you'll be easily able to answer here. 1 mark for sure from this table. Yes, sir. Fine. चलिए आगे बढ़ते हैं सर. Let's go ahead right now. Now please look at this question here. I hope you'll be able to solve that right now. Breastfeeding is contraindicated in which of the following? गैलेक्टोसीमिया, CMV, हेपेटाइटिस बी, वेस्ट नाइल सिंड्रोम. अभी अभी तो मैंने आपको बताया था ब्रेस्ट फीडिंग मतलब क्या होगा. Yes, milk will have lactose. Lactose will give you galactose. So in galactosemia, you should not do the breastfeeding. That is contraindicated. Yes. Now look at this diagram here, sir. A patient presented to OPD with a liver damage. The picture depicts the patient is having eye examined, and in this eye you can clearly see here, yes my dear friend, which type of cataract is this one here. Yes, remember it is the sunflower, sorry. Yes my dear friends, it is actually the sunflower cataract here. Now now, why I have given you this question suddenly here? Why I have given you this question suddenly here? Why? Because you should always not get confused between that oil drop cataract and this sunflower cataract over here, sir. Okay. See, many of the students are marking there as galactose. इसीलिए तो मैंने question यहाँ पर दिया है. Oil drop cataract मैंने आपको अभी picture बताया था. It will be something like this here. Okay, so don't look at that one and get confused. Oh, liver damage is given there and eyes they are given a cataract here. Please observe very carefully. It's not oil drop cataract. It's actually the sunflower cataract. And that will be actually due to what here, my dear friends? Yes, it is due to the copper. Are you understanding me here? I hope you're understanding me here, the difference here. So don't get confused between that oil drop cataract and the sunflower cataract. So please look at the image very much carefully here. Yes. Very good. Correct answers over there. Fine. चलिए आगे बढ़ते हैं sir. Now, so if you are observing like carbohydrate metabolism we are discussing right now. Carbohydrate metabolism discuss करें, and if you're observing there the sequence how we are following there, so as I told you I'm going to finish like every topic in this one, my dear friend. We are done with glycolysis, we are done with the Krebs cycle, and after that gluconeogenesis, glycogenesis, glycogenolysis, and then after this we're also going to do with the HMP shunt also. In this way every topic we're going to cover. And now welcome to one of the important table here. Many of the times in your exams they're asking about the glucose transporters here, my dear friend. Okay, now what is the difference between this GLUT and SGLT here? Remember GLUT will be the sodium independent glucose transporter, whereas SGLT will be the sodium dependent glucose transporter, sodium dependent one. Fine. Now GLUT 1 and 3, yes, they are present in most of the tissues in our body. So GLUT 1 and 3 is present in most of the tissues in our body. Like they are present in the brain, RBC, placenta, everywhere. So fine. Now they are concerned with the basal uptake of glucose. What do you mean by that? Yes, the brain, RBC and all, they require glucose. Any given point of time, they want glucose there. So therefore, what is going to happen, my dear friend? Therefore, what is going to happen there? Even though in your body, the glucose level is less, at that time also, GLUT 1 and 3, they have the ability to attract the glucose. That ability is known as the basal uptake of glucose. Basal uptake. Okay. Now, in this one, remember, if they are giving you like neurons पर कौन सा वाला glucose transmitter होता है, तो remember at that time GLUT 3 will be a better answer when compared to GLUT 1. Sir. That is an important point to note down here. So brain and the neuron here, I told you 1 and 3 दोनों भी होते हैं. In neuron, remember GLUT 3 will be a more better answer when compared to GLUT 1. Now GLUT 2, please remember with the number 2 here. Sir. विद द नंबर 2 इट विल बी प्रेजेंट ऑन द लिवर, और हम सबको पता है लिवर में क्या होता है, ग्लूकोज एंटर भी होता है और ग्लूकोज बाहर भी आता है. तो ग्लूकोज गोइंग टू एंटर द लिवर एज वेल एज कम आउट, सो इट इज अलाउिंग बोथ एंट्री एज वेल एज द एग्जिट ऑफ द glucose. It is allowing entry and exit, like two-way movement of the glucose, two-way movement of the glucose, present on the liver and the beta cells of pancreas. Okay, and even it is also present on the kidney also, sir. So liver, beta cells of the pancreas as well as the kidney will have this one. Now GLUT4 extremely important for your FMG exam. Multiple times they have asked in the exam, sir. It is present on skeletal muscle as well as the adipocytes here. Okay, and what is asked in your exam? Insulin dependent. It is out of all the glucose transporter, the insulin dependent one, sir. Okay, now. Then after that, my dear friend, remember GLUT5. GLUT5, please remember that even though the name is glucose transporter, it is mainly a fructose transporter. It is going to transport fructose mainly. And that is required for what, my dear friend? On the small intestine for absorption of fructose. And that is also present on the testis. Yes, it is present on the sperm cells, spermatozoa. Why? Because spermatozoa, yes, loves to taste the fructose. Fructose transport के लिए बहुत important है. ठीक है ना? Done. After that, my dear friends, remember sodium dependent glucose transporter 1, SGLT1 and SGLT2. One will be present on intestine for absorption of glucose, and SGLT2 will be present on the kidney for reabsorption of the glucose. Perfect? Yes. From many of the times, my dear friend, they'll be asking a question from over here. Is it okay? चलिए अब हम बढ़ते हैं आगे. The next concept to learn. What happens after a meal? यार ये इतना सिंपल सा क्वेश्चन है, आपने फिजियोलॉजी में पढ़ रखा होगा कि which hormone is going to increase? Of course, after consuming a meal, our glucose level will actually increase. And we have to decrease that glucose level. And which hormone is going to decrease the blood glucose level? इंसुलिन. तो इंसुलिन का लेवल बढ़ेगा और ग्लूकोगॉन का लेवल क्या होगा, कम होगा सर. इट इज गोइंग टू डिक्रीज. इट्स अ सिंपल क्वेश्चन हियर, so don't get confused over there. Whenever you're consuming any meal, glucose level will increase, and to decrease the glucose level back to normal, yes, of course, insulin will be released. This question was given in the FMGE June 2021. That is why I've included it over here. Next one, HMP shunt. HMP shunt, first you remember the another name of this one. It is also known as pentose phosphate pathway. And where it is going to take place, remember, it is going to take place in cytosol of many organs. Many organs. I will tell you how to remember that one. Now, in HMP shunt, the important thing is you are going to get like two important products here. Number one, it is NADPH, and number two will be the ribose 5-phosphate. NADPH and ribose 5-phosphate. Please be very careful. It is not NADH, it is NADPH. It is not NADH, remember it is NADPH. And another one will be the ribose 5-phosphate. Okay. Now, what is the importance of this one? I'm telling you they're important product. Remember, ribose 5-phosphate is the one which is helping you in the purine synthesis, in the de novo synthesis of purine. So therefore, it is required for the formation of your DNA as well as the RNA. DNA और RNA form करने में help करता है, purine synthesis में help करता है, ठीक है ना? Now, NADPH, remember the first thing, it is required for fatty acid synthesis. If fatty acids are forming, that is nothing but lipids, it is also required for lipid synthesis. If lipids are forming, yes, even for cholesterol synthesis. And if cholesterol is going to form, it is going to form all these steroid hormones. Steroid hormone synthesis also requires NADPH. NADPH is also required inside the RBC for removal of the free radical, to remove the free radical, fine, H2O2. And it is also required inside the neutrophil, and there it is required for oxygen dependent killing of bacteria. Oxygen dependent killing of the bacteria. We have discussed about all these concepts in detail in our regular classes. Now this is like last minute revision, so I'm just quickly telling you what is happening here, sir. Now once you are done with this one here, my dear friend, where is this HMP shunt taking place? It's very easy right now. Why? Please look at my important product number one, sir, steroid hormone. So wherever steroid hormones are forming in our body, all those places HMP shunt will take place. Adrenal cortex, number two, in the testis, in the ovaries, and in the placenta. So wherever, my dear friend, the steroid hormones are forming, their HMP shunt is going to take place. Will HMP shunt occur in RBC? Yes. Will HMP shunt occur in neutrophil? Yes. Okay. So in this manner, if you remember this important product here, from here itself we can actually learn from which all organs, my dear friends, yes, HMP shunt is going to take place. Fine. So please remember this very carefully. And in the very beginning of the carbohydrate metabolism, already मैंने आपको बता रखा है, what is the rate limiting enzyme here? It is glucose 6 phosphate dehydrogenase, G6PD. G6PD is the rate limiting enzyme over here. So, fine. Badhiya. So in this manner, my dear friends, yes, we are done with this topic also. Now, one of the question related to your HMP shunt over here, cytochrome P450 is associated with NADH, NADPH, FADH2 or ADP. Please remember, it is associated with your NADPH, the phosphorylated form over there. Sir. Fine. So, my dear friends, by this we have discussed all the topics regarding the carbohydrate metabolism. I hope you will be able to answer. And yes, of course, we are done with many clinical conditions also. We discussed about the glycogen storage diseases as well as the galactosemia, classical galactosemia, essential fructoseuria, and then the fructose intolerance also. Many clinical conditions are also completed here, sir. Fine. So by this, we have revised the entire carbohydrate metabolism perfectly here. I hope you are able to revise all the things. आई थिंक आप सब लोगों ने ऑलरेडी रेगुलर क्लासेस में बायोकेमिस्ट्री पढ़ाता होगा. दैट इज द रीजन वाई जस्ट बिफोर द एग्जाम, like just 1 month before your exam, I am conducting this session of Biochemistry LMR over here so that you will be able to revise the entire things. Okay. Of course, this video will be available here on YouTube. It will be available there on my YouTube channel, Dr. Azam Lectures. And you can obviously watch that n number of times. Yes. And please do watch that one day before going to the exam, sir. Just one day before going to the exam. On 2x speed, you try to watch that one. It's very much important because you're observing here. It's not like I'm telling you some haphazard points here and there. I'm not telling you any just points to mug up. We are doing like topic by topic. The entire biochemistry will be done here, sir. So please remember this one here. So revise this before going to the exam. One day before going to the exam is must revision. Yes, put it on 2x and revise this for my dear friends. Okay. So this is what I can actually do for you. I'm actually not answering the things which you are writing there in the comments over there, my dear friends. Why? Because let's focus on the studies right now. बिकॉज द टाइम इज वेरी लेस और आप सबको पता है रूल्स एंड रेगुलेशंस कैसे चेंज हो रहे हैं एफएमजी में. द थिंग्स आर गोइंग टू बिकम लाइक मोर एंड मोर डिफिकल्ट. they are becoming very much difficult internship को लेकर and all. So let us focus on studies right now. I will try to answer your questions later on. We can connect later on, my dear friends. So let us just focus here, and you also completely focus here. Don't get distracted here and there, and do this one here, sir. Fine. Next one. Now after that, let us start with the proteins topic right now. Proteins, हम सबको पता है, they are polymer of amino acid. In proteins topic, let us quickly revise all the 20 amino acids with their classification. 20 amino acids and their classifications. The classification number 1 is based on the structure. Now, based on structure, you'll be having the aliphatic amino acid, glycine, alanine, valine, leucine, isoleucine. Glycine, alanine, valine, leucine, isoleucine. Out of that, please remember, my dear friend, the simplest amino acid will be the glycine. The smallest amino acid will be the glycine. Simplest and smallest one. And out of this, please remember, valine, leucine, and isoleucine, these three together will be known as the branch chain amino acid. Branch chain amino acid. Now, after that, OH group containing amino acid, hydroxyl group containing will be serine and threonine. Sulfur containing amino acid will be cysteine and methionine. Acidic amino acid will be aspartic acid, glutamic acid, asparagine, and glutamine. Basic amino acid will be lysine, arginine, histidine. Aromatic amino acid, very much important one, tyrosine, tryptophan, and the phenylalanine. And amino acid will be the proline. Okay, so I'm just going quickly through the names of this one here. Why? Because I hope you all know all the amino acids very well. So एक बारी ये ना refresh हो जाएगा आपके mind में. Now once it is refreshed in your mind, let us see the second classification based on nutritional requirement. Essential, semi-essential, and the non-essential. Essential amino acid, how to remember that one? Please remember TV till 8 p.m. And 8, why? Because there are totally 8 of them which are essential here. Fine. TV till 8 p.m. sir. Now T here stands for number 1, that is your tryptophan. V for valine. And then till T stands for, yes, threonine. I for isoleucine. L for leucine. L for lysine. And then 8 p.m. P stands for phenylalanine. M stands for the methionine. Sir. TV till 8 p.m. And semi-essential amino acids are only 2 of them. One is your arginine and histidine. But in one of your exam, both the options were given. When both the options are given, remember, arginine will be a better option, sir. And after that, please don't remember the other one. All the rest, all amino acid will be the non-essential amino acids, sir. Non-essential amino acid. Now, next one is based on the metabolic fate. Some of the amino acid will undergo metabolism to form ketone bodies, ketogenic amino acid. Some will undergo metabolism to form glucose, glucogenic amino acid. And then some will form both glucose and ketone body, glucogenic and ketogenic amino acid. Now out of that remember ketogenic, both leucine and lysine sir. ये कैसे होगा? मैंने आपको अभी अभी बताया था when I revised gluconeogenesis, formation of glucose from non-carbohydrate sources. I told you that how to remember the examples of non-carbohydrate sources. LPGA, A for amino acid. All amino acids are going to form the glucose except two of them, leucine and lysine. Why? Because these two will be the ketogenic amino acids. Ketogenic one. And agar aapko yaad rakhna hai, which is more better option here, sir? Remember, leucine will be like more ketogenic here, sir. Leucine is more ketogenic when compared to the lysine. Now, both glucogenic and ketogenic, my dear friend, they will be aromatic amino acid. Tyrosine, tryptophan, and phenylalanine. Along with that aromatic, remember isoleucine. Some students love to remember with the mnemonic here as PITT, PIT here. P for phenylalanine, I for isoleucine, T for tyrosine, T for tryptophan. And then rest all will be the glucogenic, no need to learn them. Is it okay? Now, apart from this 20 amino acid, please remember the 21st amino acid, that is selenocysteine. Now, two MCQs to remember here. MCQ number 1, selenocysteine will be formed from where? Please remember it is formed from serine or serine, whatever you love to say. Serine is going to form the selenocysteine. Please remember, when it was asked in your exam, some students have answered like selenium, some students have answered like cysteine. Please remember, it is forming from serine. And what is the codon which is going to code for selenocysteine, 21st amino acid? I hope you remember the three stop codons. UAA, UAG and UGA. Out of the three of them, remember UGA is going to code for selenocysteine. And for 22nd amino acid, remember the name, it is pyrrolysine. And pyrrolysine, remember the only and only one thing to remember here is the codon. The codon for pyrrolysine will be UAG. That is also one of the stop codon. Please remember this already, that has been tested in your exam, my dear friends. I hope this is very clear here. Next one. Now, nowadays in your FMG exam, a trending concept, transamination. Trending concept. You can expect one question from here in your January exam, sir. Trending concept. Every exam they're giving one question from here. Now, my dear doctors, remember, whenever you're going to consume the proteins, okay, all that non-vegetarian food and all, consume the proteins. Proteins are made up of amino acid. So indirectly, we are loading our body with ammonia. Now, when ammonia is coming in our body, tell me ammonia is a good substance or a bad substance? It's very bad. So because ammonia is like very bad substance, when hyperammonemia will occur, you know very well it is going to affect our brain. It will lead to cerebral edema. So therefore, we need to eliminate that one. We need to remove that one. But how to remove that one? Ammonia is so toxic substance that even it cannot be transported in our body in the form of ammonia. Ammonia is so toxic substance that in our body, it can't even be transported in the form of ammonia. It will be transported in some other form. So that form you have to remember. Did you understand the background story? So you have to remember the form in which ammonia will be transported. Is it okay? So my dear friend, from most of the tissues in our body, ammonia is going to the liver in the form of glutamine. It is going in the form of glutamine. Now, in your MC exam, they have asked specifically from the brain. From the brain, it is going to the liver in the form of glutamine. And then remember, from the skeletal muscle, the ammonia will go to the liver in the form of alanine. And I taught you about this one in Cahill's cycle. Which is also referred to as the glucose alanine cycle here, sir. So we have learned about that one in the Cahill cycle. I hope you are very, very clear with this one. So my dear friends, remember from this picture, definitely one question is going to come in your exam. They're asking repeatedly in your exams, my dear friend. तो एक बार दोबारा repeat करते हैं. It is like worth repeating. It is worth repeating, my dear friends. From most of the tissues in our body, ammonia is going to liver in the form of glutamine. And from the brain, the ammonia is going to the liver in the form of glutamine only. But remember, from the skeletal muscle, it is going to the liver in the form of alanine. Okay. Next one. Now, after this ammonia is coming to the liver, in the liver, what is going to happen? That ammonia will be getting converted to urea by urea cycle. The urea cycle, first you remember the another name. It is also known as the ornithine cycle, and it is also known as the Krebs-Hanslade cycle. Sir. Urea cycle or ornithine cycle or Krebs-Hanslade cycle. First, where is urea cycle taking place? In the liver. But remember, the reactions are taking place both in the cytoplasm as well as mitochondria also. Then number 2 point that you have to remember here after the location is, yes, name is done, location is done. Now let us see the cycle here. It's a very small cycle, hardly like, 5 reactions in this one. Now look here, starting from here, ammonia will be first of all combining along with the carbon dioxide to form carbamoyl phosphate. That is why the name of the enzyme will be carbamoyl phosphate synthase 1, CPS1, sir. Carbamoyl phosphate synthase 1, that is CPS1 here. Now this carbamoyl phosphate is going to combine along with citrulline, and together they are going to, carbamoyl phosphate is going to combine along with ornithine, and together they are going to form citrulline. Now citrulline is going to combine along with aspartate, and together it is going to form arginosuccinate. And arginosuccinate will actually break down to form now arginine with the release of fumarate, and arginine will actually break down to form. Finally, it is going to give rise to urea. And again, it is getting converted towards the ornithine back. Understood. So this entire cycle in our regular classes, we have learned that in the class itself, like 5 reactions are taking place here. Now, out of these 5 reactions, what you have to remember here is question number 1. What is the formula of urea? NH2CONH2. The formula of urea will be NH2CONH2, sir. In this, the first NH2 group is obtained from ammonium ion that you are adding in the reaction number 1, ammonium ion, जो आपने यहाँ पर ऐड किया था. And then CO group will be obtained from carbon dioxide that you have added over here. And then, my dear friend, one more ammonium ion will be added by aspartate. It is added by aspartate here, sir. Okay. So in your exams, they're also asking about the sources of urea, sir. So urea formula will be NH2CONH2. The first NH2 group is coming from ammonium ion. The CO group is coming from carbon dioxide. And then the one more NH2 group is actually coming from the aspartate, my dear friends. Now, once you are clear with this one here, the second question is about the rate limiting enzyme. So which one will be the rate limiting enzyme here? CPS1. This one here will be the rate limiting enzyme of the urea cycle. And lastly, I told you like it is taking place both in cytoplasm as well as mitochondria. So remember, out of all the reaction, the first reaction and the second reaction will take place in the mitochondria, whereas the remaining 3 reactions will take place in cytoplasm. In this manner, we have covered like all the points regarding the urea cycle has been, yes, covered here, sir. Now moving ahead, after completing this ornithine cycle, the next one, another important concept from where the questions will come in the protein metabolism. Okay, my dear friends, listen with all your ears open right now. 2 things you have to remember here: amino acid and what is the important product obtained from there. And number 2, you have to remember about the diseases associated. I'll give you like 4 diseases here, and the questions will come. Definitely one question is going to come from the, yes, one of the 4 diseases there, guys. Okay, now first let us discuss about the amino acid and what is the important product obtained from here. Tyrosine. Tyrosine is the one which is going to form number 1, melanin. It is going to form this pigment melanin here, as well as T3, T4 here, as well as catecholamines. You know the catecholamines very well. Number 1, there is dopamine here, then norepinephrine here, and then there will be epinephrine here. All of them are derived from where? From the tyrosine amino acid. Tryptophan is going to form melatonin. Observe the way I have highlighted here: tryptophan, melatonin, serotonin. So tryptophan is the one which is going to form your melatonin as well as the serotonin, my dear friends. And then remember, it is also going to form the vitamin B3, very much important. In fact, they have asked that, 60 milligrams of tryptophan is going to actually form 1 milligram of niacin, that is vitamin B3. So niacin is also derived from where? Tryptophan. It is very, very much important. Clear? Now, once you are done with this one here, next one is histidine. Histidine is going to form what here? Histamine. But in the exam, the question was, histidine is going to form histamine by which reaction? Remember, it is by decarboxylation. That is by decarboxylation. And the repeat question which is asked in your exam is about the arginine. Arginine is the one which is going to form nitric oxide, NO, and it is going to form nitric oxide in the presence of an enzyme, that is nitric oxide synthase, NOS. Okay. And finally, glycine, another important thing for your FMG exam. Glycine is the one which is going to form your heme. Heme synthesis in fact begins with glycine. अगर आपको याद होगा, succinyl CoA combines along with the glycine to form delta ALA synthase enzyme, है ना? तो heme synthesis begins from glycine. Number 2, heme is also required for purine ring formation. I will show you the entire purine ring just now within few minutes. Don't worry about that. And it is also required for the formation of glutathione. So remember, glutathione is a tripeptide. Glutamate. Thio is the sulfur containing amino acid, that is cysteine, and then ONE1, one glycine. So you have to also remember the three amino acids here, sir. So number one, it is going to form from glutamate as well as sulfur containing amino acid, that is cysteine, and then the first one, that is your glycine, sir. And then remember, yes, glycine is also required for the formation of creatine, and creatine formation is again done by the three amino acid. You have to remember, like GAM. G stands for glycine, A for arginine, M for methionine. So basically, we are learning all the important products obtained from which amino acid. Questions can be coming based on this one here. Now, once you are done with this one, amino acid and special product, can you please answer this question for me here? Can you answer this question here, my dear friends? Melanin will be synthesized from which of the following amino acid? Yes, of course, it will be synthesized from the tyrosine. So melanin will be synthesized from where? Tyrosine. Absolutely correct. Similarly, nitric oxide, that is NO, will be synthesized from arginine. So these type of questions will be coming in your exam. Which amino acid is going to form which important product is very much important here. Now, after that, I told you just now, T4 and T3, T3 and T4 are formed from tyrosine. Well and good. But what is the question that they are asking in the exam? Peripheral conversion of T4 to T3 will occur by which enzyme? Now, my dear doctors, what is the meaning of T4? T4 is nothing but the tetraiodothyronine. So it is having how many iodines here? You can clearly see the 4 iodines here. That is your T4. And that has to be converted to T3. That means what we have to do here? We have to remove one iodine from here. Only we are remaining with 3 iodine here. And we are going to remove the iodine present on 5' position here. So from the 5' position, if you remove that iodine there, my dear friend, the name of the enzyme here will be obviously deiodinase. You know very well in biochemistry, whenever you're going to remove something, it will be D. So deiodinase, and remember it is 5' deiodinase. We are going to remove the iodine from the 5' position. Then only what is going to happen, my dear friend, this is going to be the active form of the T3. Active form of the T3. Now suppose if you are going to remove the iodine from the 5th position, that is your 5th carbon here. अगर आप 5th carbon से यहाँ से remove करोगे, तो therefore what is going to happen? What is the name of the enzyme? That is 5-deiodinase. It is not 5-deiodinase, it is 5-deiodinase. And please remember, there is the inactive form of the T3 which is known as the RT3. Reverse T3 कहते हैं, and reverse T3, this one will be the inactive form. So whatever, the peripheral conversion of T4 to T3 will be done by which enzyme? Deiodinase. It is not thyroperoxidase. Please remember, it is deiodinase. Okay. Thyroperoxidase is the one which is actually helping you in, yes, oxidation of iodide to iodine. Oxidation of iodide to iodine, so not removal. Removal तो बड़ा आसान है यार, नाम में ही लिखा है. It is there in the name itself. From 4 iodine we are getting like 3 iodine. So 1 iodine will be removed from there. Deiodinased. Done. After that, one more thing which is asked in your FMGE exam is about the methylation reaction. Now in this diagram you can clearly see this is norepinephrine. Norepinephrine will be getting converted to epinephrine. And how it is getting converted, norepinephrine to epinephrine? You can clearly see here the methyl group will be added there by addition of this methyl group. Therefore it is methylation. Fine. But now the important question, the $1 million question here is that who is going to add that methyl group? The methyl group is added by SAM. And what is the full form of SAM here, sir? S-adenosylmethionine. That is the one which is going to be the methyl group donor. That is what they're asking in your exam, sir. Very nice. Okay, so norepinephrine to epinephrine is one of the examples of methylation. But why I am including this one in our LMR session here? I want you to remember the SAM here, sir. SAM is the methyl group donor that is going to add the methyl group over here, sir. Perfect. There's also important thing to revise before going to the exam. Done. Now after completing all these points here, like from the amino acids, we are getting like which important product that is done. Now I am going to tell you about the 4 diseases here, sir. Phenylketonuria, alkaptonuria, and then the maple syrup urine disease and Hartnupp's disease. And from these 4 diseases, one question will come. I hope all the students who are listening to me very, very carefully here, my dear friends, all my dear students who are listening to me carefully here, please remember and note down wherever I'm telling you, like one question will come from where. Because those segments you have to, learn. Those segments, like from these 4, as a student, you have to remember that if I learn these 4 diseases, okay, I'm getting like one question from here. If I learn the important products from amino acid, one question is possible from there. So learn in that manner. Don't just randomly learn anything. Okay. चलिए start करते हैं सर. Now, phenylketonuria is occurring due to deficiency of which enzyme? Phenylalanine hydroxylase. अब यार ये एंजाइम हम कहाँ पर पढ़ते हैं? Where do we study this enzyme? Remember, phenylalanine is going to get converted to tyrosine. It is going to get converted to tyrosine, and this will occur by addition of OH groups. Now you know very well what is the name of that OH group, hydroxyl group. And because we are adding the hydroxyl group, the name of the enzyme here will be hydroxylase. Add the name of the substrate here, phenylalanine hydroxylase. Lovely. Easy to make the name of the enzyme. And now suppose if this enzyme is deficient, obviously what is going to happen? Phenylalanine levels will increase and it will start excreting in the urine. That condition is known as phenylketonuria. Phenylketonuria. Okay. अब इसमें ना features क्या होते हैं? What are the clinical features? Now common sense is that if this reaction does not occur, tyrosine will not be formed. और अभी अभी मैंने आपको बताया था tyrosine is the one which is going to form melanin. Okay. If there is a decreased tyrosine, decreased melanin in the skin, that will be hypopigmented skin. That skin will become pale skin here, sir. And after that, if the melanin decreases in the hair, that will be like blonde hair, sir. And after that, yes, my dear friend, I told you that tyrosine is also going to form T3 and T4 also. If thyroid hormones decreases, I hope all of you have learned in physiology that thyroid hormones are necessary for what? They are necessary for proper development of brain also. And if the brain is not developing properly there, obviously it will lead to what? Mental retardation. So intellectual disability, microcephaly and seizures. Brain is not developing properly. Yes, that is explained by T3 and T4. Apart from that, there will be a characteristic mousy or musty odor. Mousy or musty odor. Okay. Yes, Dr. Shadab, albinism, it's not due to this one here. Albinism is due to defect in the tyrosinase enzyme. टायरोसिनेज एंजाइम दैट इज अ डिफरेंट वन ओके नो टायरोसिनेज एंजाइम से होता है तो दोनों में कंफ्यूज मत हो जाइए डोंट गेट कंफ्यूज्ड इन बोथ ऑफ देम ओके आई होप योर डाउट इज क्लियर हियर that is about the phenylketonuria that you have to remember. Easy to remember all the clinical features with the help of the way I told you here. If this enzyme is deficient here, my dear friend, there will be no tyrosine. If there is no tyrosine, melanin will be decreased there, and if it decreases melanin, then what is going to happen there? Yes, hypopigmentation as well as the blonde hairs. And if there is no T3, T4, then what is going to happen there? Intellectual disability, microcephaly, seizures, and all those will occur. Fine. One disease completed here. Please remember that one. Now, alkaptonuria or black urine disease. What is the problem here, sir? The enzyme deficient here will be the homogentisate oxidase. Okay. Homogentisate oxidase enzyme will be deficient here. Homogentisic acid level will increase. That will get converted to benzoquinone, and finally that is going to form ochronotic pigments. Okay, now what is going to happen? This will be going out into the urine. Collect that urine sample and keep that urine sample for some time, and you will observe that the color of the urine changes to black color or coke color. Black urine disease or alkaptonuria. And similarly, that will also get accumulated in the connective tissues in our body like cartilages and all, and that you can clearly see in the pinna. Here you can see the elastic cartilage will be there, and that is getting converted to blackish in color. And accumulation in the sclera, here you'll be able to see the black colored patches in the sclera over there. So by that condition is known as what, guys? Ochronosis कहते हैं sir इसको. This one here will be known as ochronosis. I hope this is one done here. So alkaptonuria or black urine disease, yes, it is due to deficiency of which enzyme? Homogentisate oxidase, already asked in your exam, sir. The urine is getting converted to black in color on long standing. Please remember that word, on long standing. That means we are not going to do any reaction over there. Just keep that for some time. It will get converted to black color or coke color, sir. Perfect. Welcome to the disease number 3. That is maple syrup urine disease, sir. I hope you'll be able to easily answer this question over here. A child presents with the complaints of urine turning dark on standing. It is caused by deficiency of which enzyme? Yes, the name of the enzyme will be homogentisate oxidase, asked in FMGE June 2022. FMGE June 2022. Maple syrup urine disease. अब यार ये maple syrup urine disease will be occurring due to what? I hope I told you just now about the branch chain amino acid. There'll be three of them. One will be the valine, another one will be the leucine, another one is the isoleucine. Now, when this valine, leucine, and isoleucine, these three branch an amino acid, when they undergo metabolism, and finally they are going to form certain product there. We are not concerned about that right now. Now, please remember one of the enzyme during that metabolism. Okay, now one of the enzyme here will be branched chain because they are branched in amino acid. Keto acid. Yes, I hope you all know very well. From the amino acid, if you remove the ammonia, then what will be remaining? The keto acid, dehydrogenase. Branch chain keto acid, dehydrogenase. And I remember this one here as BCKDH. Branch chain keto acid dehydrogenase. Branch chain keto acid dehydrogenase. Okay, now this enzyme, if it is defective, remember it will lead to what, guys? Maple syrup urine disease. So maple syrup urine disease occurs due to which enzyme deficiency? Yes, branch chain keto acid dehydrogenase. Okay, now my dear friend, what is going to happen in this one? Why the name is given like maple syrup there? Why? Because urine smells like maple syrup. And what is the smell? The smell is like burnt sugar. Please remember that. The urine smells like burnt sugar over there. Are you clear with this one here, sir? So in this manner, you are able to clearly remember about the maple syrup urine disease. Now the next one and the last one, the fourth one which I want you to remember is about Hartnup disease, given recently in the July exam, sir. Last July exam it was given, 2024. Now in Hartnup disease, what is going to happen? There will be decreased absorption of amino acid from intestine. Decreased absorption of amino acid from intestines. Okay, because the receptor, the channels which are going to absorb that one, absorb the amino acids will be actually defective here. Fine, but mainly which amino acid will be decreased here in Hartnup disease? That is tryptophan. Tryptophan here, sir. Fine. ट्रिप्टोफैन हियर, अगर ट्रिप्टोफैन का कमी होगा, अभी अभी मैंने आपको बताया था ट्रिप्टोफैन, इट इज गोइंग टू फॉर्म व्हाट सर? नंबर वन, इट इज गोइंग टू फॉर्म मेलेटोनिन. Number two, it is going to form serotonin. And number three, it is also going to form what, sir? Vitamin B3. Okay, niacin भी form होता है. Now, common sense. In Hartnup disease, if tryptophan is not able to be absorbed, then therefore there will be no formation of vitamin B3. अगर vitamin B3 form नहीं होगा, तो obviously it will lead to pellagra-like symptoms. Pellagra-like symptoms. So in Hartnup disease, what is going to happen? There will be pellagra-like symptoms, the 3D, dementia, diarrhea, and dermatitis. You know that very well, guys. Clear? So that's all. So by this, I have completed like all the 4 diseases here, my dear friends. So please remember, like definitely one question will come from all that 4 diseases. Clear? Next one. By this, I have completed about the protein metabolism, entirely about the amino acid classification. We have seen about the important products obtained from there, as well as all the diseases also. Questions will be basically coming from here. Urea cycle and all those things, transamination is also done, guys. So everything regarding the protein metabolism is completed here. Just watch the video as many times as possible before going to exam. It is surely going to benefit, sir, because you're not leaving any topic over here. Fine. Let us go to the lipid metabolism right now. Now the first question here, what is the neutral lipid which is stored in the liver? Now the options which are given here, triacylglycerol, cholesterol, phospholipid and the fatty acid. Which one will be the neutral lipid present in the stool? Now what is the meaning of neutral lipid, sir? Look here. Lipid we are going to divide into 2 categories here. One will be the amphipathic lipid and another one is the neutral lipid. Amphipathic lipid मतलब ये क्या होगा? It will be polar. It will be having the polarity. Now you can clearly see here, one of the example is cholesterol. Why? Because it's having that free OH group here. Another example will be the phospholipid. Why? Because phospholipid will have that negative charge on that one. So because it is having the negative charge there, the free OH group available there, that's the reason why this is known as amphipathic lipid. Whereas neutral lipid, मतलब क्या होना चाहिए? There should be no like free functional group available. Okay, so therefore one of the example for this one is cholesterol ester. इसका मतलब क्या होता है? You can clearly see the same structure of cholesterol here. In the place of that OH group, what we have done here, we have added a substance over there. That means normally we are going to attach a fatty acid over there. So cholesterol ester बन जाएगा once you attach over there. Now there is no more OH group, free OH group available there. So it is not at all like polar. It is like non-polar. Neutral one. And of course, the very famous one, TAG, triacylglyceride. or simply triglycerides. There will be one glycerol molecule, and on this one glycerol molecule there will be like three fatty acids attached. तभी तो इसको क्या कहा जाता है, triacyl glycerol कहा जाता है. And that is also like the neutral lipids. You have to remember the examples of this one. Now welcome back here, my dear doctors. The neutral lipid which is stored in the liver will be the triacyl glycerol. This is a neutral lipid. Cholesterol, phospholipid. Yes, they are not the neutral lipids over there, guys. Fine. Are you clear? चलिए सर आगे बढ़ेंगे. तो दिस लाइक द इंट्रोडक्शन ऑफ द लिपिड्स हियर. तो वी आर डन विद द क्लासिफिकेशन हियर. नाउ इन द लिपिड मेटाबॉलिज्म, yes, questions will be coming from here. Always what I do here is divide the lipid metabolism into these two categories here. One will be about the cholesterol. One is about the cholesterol, and another one is actually about the fatty acids. Cholesterol synthesis and degradation and fatty acid synthesis and degradation, guys. Okay, now cholesterol synthesis, please remember the only and only point that you have to remember here is, yes, what is the rate limiting enzyme of cholesterol synthesis? The rate limiting enzyme of the cholesterol synthesis will be HMG-CoA reductase. HMG-CoA reductase is the rate limiting enzyme. Okay, and one more point. Yes, already मैंने आपको बता रखा है कि what is the starting substance for cholesterol synthesis? Of course, it is acetyl CoA. Acetyl CoA is the substance from where the cholesterol synthesis is going to begin. Next one, fatty acid synthesis. Yes, what is the rate limiting enzyme of this one? Acetyl CoA carboxylase. What is the point to remember? This requires biotin as a coenzyme. मैंने आपको क्या बोला था, carboxylases के लिए क्या चाहिए? What do you require for carboxylases enzyme? Which coenzyme? Biotin coenzyme is required there. And again the same thing, fatty acid synthesis will also begin from where? Acetyl CoA only. Now after that, beta oxidation of fatty acid. This is about degradation right now, sir. Now beta oxidation of fatty acid, what is the rate limiting enzyme? That is CAT1, carnitine acyl transferase 1. CAT1 will be the rate limiting enzyme here. And finally ketogenesis, it will be HMG-CoA synthase. Ketone body formation, HMG-CoA synthase. So these are all the pathways in the lipid metabolism, and you have to remember their rate limiting enzyme here, sir. Fine. Now please answer this question here, sir. Cholesterol synthesis is from where? I told you just now it begins from acetyl CoA. Acetyl CoA. So just remember 2 points here for your FMG exam. That is more than enough about the cholesterol synthesis. Yes, what is that? The beginning starting substance will be acetyl CoA, and what is the rate limiting enzyme? HMG-CoA reductase. That's all. And that's all, my dear friend. Cholesterol synthesis, they'll be asking you a question from these 2 points only. That's all. Now, what about the degradation of cholesterol? What about the degradation of cholesterol, my dear friend? Remember, cholesterol will undergo degradation to form all the steroid hormones present in our body. Even this is also one of the previous year question already asked in your exam. Steroid hormones are formed from where? They are formed from cholesterol. Cholesterol is the one which is also going to form your vitamin D, and cholesterol is going to form bile acids. That's all. So remember these 3 points in the degradation of cholesterol. This topic is done. Okay, now. So remember the 2 points in the cholesterol synthesis, and remember these 3 points in the degradation of cholesterol. Done. This topic is completed here, sir. Now moving ahead with the next one. As I told you, in every topic, I am telling you about the clinical correlations over here. So here also, welcome here, my dear friend. In the lipids topic, questions will be coming from where? The lipid storage diseases. In Niemann-Pick disease, what is the enzyme deficient? Sphingomyelinase. Similarly, the very important one, Gaucher's disease, the enzyme deficient will be beta glucosidase, or the complete name will be beta glucocerebrosidase. And in the Krabbe's and Fabry's disease, you can clearly see here, it is galacto and galactocerebrosidase. Okay, remember Fabry's, it will be alpha. Fabry's, it will be alpha. And in Krabbe's, remember it is beta galactocerebrosidase. Remember in this manner. And after that, the Tay-Sachs disease is due to deficiency of hexosaminidase A. And finally, the Sandhoff's disease will be due to deficiency of which enzyme? Hexosaminidase A and B. Fine. So we have discussed about all these things in detail in our regular classes, and in this LMR, please quickly revise the enzyme deficient and the disease which is related over there, guys. Again. Now, after completing this one here, please look at this one, the next clinical correlation about hypercholesterolemia. Yes, hypercholesterolemia. A 25-year-old male presents with hypercholesterolemia. His elder brother also died of the heart disease a few years back. Okay, there is a family history also. LDL receptors are defective here. LDL receptors are defective here. I hope you all have watched my, biochemistry classes. In the regular classes, we have seen that the LDL, HDL, VLDL, all the lipoproteins I explained you in detail. And in that, I explained you the entire topic just by simple two diagrams. One diagram is about the exogenous fat, the fat that we are consuming from outside, and one was about the endogenous fat, the fat that is going to be produced inside our body only. Okay, so in that I told you about the LDL, low density, the LDL receptors. Yes, my dear friends, you're absolutely right there. The familial hypercholesterolemia, that is the right answer, where you have LDL receptor defect in that one. Is it okay? So now let me show you here, my dear friends. Hypercholesterolemia. In hypercholesterolemia, remember there will be the findings of tendon xanthomas, accelerated atherosclerosis will occur in this one here. Tendon xanthomas, please look at the pictures very carefully there. There'll be accumulation of the fat over there. Tendon xanthomas will occur, as well as there'll be accelerated atherosclerosis will occur. Now for hypercholesterolemia, example here, the familiar hypercholesterolemia. And it is going to include which types? I hope you know the different types of this one. There'll be type 2A, which will be included over here. And what is the defect here? LDL receptors are defective here. And remember, it's an example of autosomal dominant kind of category here. Now, similarly, what is the difference with the hypertriglyceridemia? Triglycerides will be elevated in this one. The examples here will be the eruptive xanthomas. You can see the small xanthomas over here. That is the eruptive xanthomas over here. And recurrent pancreatitis as well as the lipemia retinalis. Now, this will be including which types over here? Triglycerides are elevated in which type here? The type 1, type 4, and type 5. Cholesterol elevated here in type 2A, that is familial hypercholesterolemia, whereas triglyceride will be elevated in the type 1, type 4, and type 5. Clear? That is about the hypercholesterolemia. Fine. Now, which of the following pathway take place only and only in the cytoplasm? Now, why I have included this question over here? If you are along with me, observing all the things very, very carefully, my dear friend, till now, I have completed carbohydrate, protein, and lipid metabolism is also completed. So let us summarize that one here. So which is taking place only in the cytoplasm? Of course, glycolysis will occur only in cytoplasm. Beta oxidation of fatty acid. Please remember, beta oxidation will be taking place in mitochondria. That should be your answer. And the TCA cycle or Krebs cycle will also occur in the mitochondria, whereas urea cycle will take place in the cytoplasm as well as mitochondria. So are you understanding my intention here? Why I have given you this one here? Yes, my dear friend. Yes. After completing carbohydrate, protein, and lipid, you can actually have a comparison and you will be able to answer here. Perfectly done. So by this, all the three important metabolisms are completed. Now welcome to the fourth one, nucleotide metabolism. What I'm observing in this topic is always there is like one question coming from this topic in your MCA exam. If you are able to recall the July exam question, in the July exam, there was a question from Lesch-Nyhan syndrome. Lesch-Nyhan syndrome. Always they're asking like one question from the nucleotide metabolism also. Fine. Now in the nucleotide metabolism, first thing to remember is about the purine ring and the pyrimidine ring. Now remember, my dear friend, the purine ring will be the double ring structure. There are two rings here, you can clearly see. But one of the ring will be the hexagonal shape, whereas the another ring will be the pentagonal shapes. And whereas the other side, you can see the pyrimidine there, the pyrimidine will be actually only a single ring structure. And apart from this one, remember the purine ring will be heterocyclic. Homo means the same, hetero means the different. So it is made up of like different atoms over here. Heterocyclic here, sir. Now you people actually have to remember like which carbon and which nitrogen will be contributed by which substance. Sometimes in your FMGE exam, they're asking the question from there. So please remember nitrogen number 1 will be contributed by aspartate. Carbon number 2 and carbon number 8 will be contributed by tetrahydrofolate. And then nitrogen number 3 is contributed by, and nitrogen number 3 as well as 9 also, it will be contributed by glutamine. And then carbon number 4, carbon number 5, nitrogen number 7. These three will be contributed by glycine. Okay. Now welcome to the pyrimidine ring here. In the pyrimidine ring, this nitrogen here, you can clearly see it is contributed there by ribose 5-phosphate. Then this carbon here is by carbon dioxide, and this nitrogen here by glutamine. And remaining 3 carbons over here are contributed by aspartate. I hope this is like very, very clear here. So basically you have to remember about the sources, my dear friends, here. That is all about just learning and memorizing the structure there. Okay. Next one. Now welcome here. We have to learn two things here, my dear friend. Number one is the purine synthesis, and number two will be the purine degradation. Now in purine synthesis, remember, it will be occurring by two methods. One will be the de novo synthesis of the purine, another one will be the salvage pathway. In de novo synthesis, the starting substance ribose 5-phosphate. Try to recall, I told you, it is obtained from HMP shunt. I hope you remember the two important products in HMP shunt. One is NADPH, another one is the ribose 5-phosphate. And NADPH and ribose 5-phosphate. So HMP shunt से हमको ये मिलता है. Now this ribose 5-phosphate is getting converted to PRPP, that is phosphoribosyl pyrophosphate. Phosphoribosyl pyrophosphate. That is why the name of the enzyme here will be the PRPP synthetase. PRPP synthetase. And after that, in the next reaction, what is going to happen? Glutamine will come here and it is going to transfer the amine group. That is why it is going to become phosphoribosyl amine. And because we are transferring the amine group here, the name of the enzyme will be amidotransferase. And you know the way how to make the full name of the enzyme. Add the name of the substrate over there, sir. Amidotransferase. And this one here will be the rate limiting enzyme, sir. Done. So in this manner, you're able to recall here de novo synthesis. What is the starting substance? Ribose 5-phosphate. What is the rate limiting enzyme? Amidotransferase. Just these two reactions you have to remember here. After that, it will undergo all like many reactions to form your IMP, inosine monophosphate, and that itself is going to form your AMP, adenosine monophosphate, as well as the guanosine monophosphate, sir. AMP as well as the GMP there. Perfectly done here, sir. Is it okay? And after that, in the salvage pathway, that is very much important, you will get one very important enzyme here. The name of the enzyme is HGPRT. Okay, HGPRT is hypoxanthine guanine phosphoribosyl transferase. Hypoxanthine guanine phosphoribosyl transferase. Okay. And if this enzyme is actually deficient, then what is going to happen? It will lead to Lesch-Nyhan syndrome. It will lead to what, guys? Lesch-Nyhan syndrome will occur here. Fine. So when this enzyme is deficient, it will lead to what here? Lesch-Nyhan syndrome. Now in this syndrome, you have to remember the three things here. Number one, the patient will be suffering with gout. So therefore, it is going to come to you with what here? It is going to come to you with the joint pains. Joint pains here, sir. Gout hoga. Number two, there will be like mental retardation, neurological disorder hota hai. And number three, the most important thing is self mutilation. The person will be harming himself. Self mutilation bhi hota hai. ठीक है ना? If you see a combination of these three here, joint pains, mental retardation, and self mutilation, yes, the answer is Lesch-Nyhan syndrome. Enzyme deficient is HGPRTS. Last July, this question was asked, guys. Okay. Now welcome to the next topic, that is degradation of the purine. So purines like your AMP, GMP and IMP, they will undergo degradation. You don't have to worry about the reactions in the middle here. They will never ask you in the FMGE exam. Please welcome to the last two reaction. Finally, they are going to form hypoxanthine. From hypoxanthine, we are going to get xanthine. From xanthine, we are going to get the uric acid. Okay, so please remember, my dear friend, yes, uric acid, if it is elevated here, hyperuricemia, that condition is known as what here? Gout. Now in gout, what is going to happen? There will be formation of monosodium urate crystal, MSU crystal, monosodium urate crystals, MSU crystal. And this MSU crystals, they will be like needle shaped crystal. Now this needle shaped crystals, they will be actually accumulated in the joint, and in the joints they are going to irritate the synovial membrane. And if anything is irritated in our body, what is the response from our body? That will be inflammation, and therefore what is going to happen? It will lead to itis. Joint is known as arthritis, and that is known as, yes, gouty arthritis कहते हैं हम. क्या कहते हैं इसको? Gouty arthritis कहते हैं सर. ठीक है ना? तो gouty arthritis यहाँ पर इसलिए होता है. Now, what is the treatment for that one? Yes, you know about the acute gout and the chronic gout and all, but one of the very famous drug, that is your allopurinol. Allopurinol का mechanism of action क्या होता है sir? What is the mechanism of action? Remember, it is going to inhibit xanthine oxidase. तो mechanism of action यहाँ पर क्या होता है, that is going to inhibit which enzyme, xanthine oxidase. That is why this is important here. Another very important thing, yes, has been asked multiple times in your FMG exams. Please take care about that one. Okay. Now once you are done with this one here, yes my dear friend, please look at this question here. Try to answer this question right now. Try to answer this question here. A postmenopausal woman presents with a pain in the first metatarsophalangeal joint. First metatarsophalangeal joints. This one here. Oh ho. यहाँ पर आप देख सकते हो. Now the following the intake of the alcohol, as shown in the image, the drug used should act by inhibiting which of the following enzyme? So what is the drug which is used in the treatment of gout here? Yes, allopurinol. That is going to inhibit which enzyme? Xanthine oxidase. That is how the question was asked in your FMG January 2023. FMG January 2023. Clear with this one here? So by this, we are done with the synthesis of purine and we are done with the degradation of the purine. Yes, my dear friends, please remember that is the reason why, yes, one question will be definitely asked from here. That is the reason why this topic is very much important here. Take it now. Next one, sir. Now the next topic that we are going to discuss, enzymes topic. 1 mark for short from this enzymes topic, sir. Every time they'll be asking in your exam. There'll be definitely one question coming from the enzymes topic here. Now let us see the questions will be coming from where. So my dear friends, remember in enzymes topic the question will come from enzyme inhibition. लेकिन enzyme inhibition पढ़ने से पहले, before learning about the enzyme inhibition, you have to know about these two graphs here, sir. The first one here is the Michaelis-Menten graph. Michaelis-Menten graph is giving you the correlation between the substrate concentration here and the velocity. And we know that the concentration of the substrate and the velocity are directly proportional to each other. So as you increase the concentration, yes, as well as what is going to happen, the velocity keeps on increasing there. So therefore you are going to get a parabolic graph here. Why? Because as the substrate concentration increases, velocity keeps on increasing proportionately. But here the most important thing is, what about the Michaelis-Menten constant, Km? Km is nothing but Michaelis-Menten constant. Now, what is the definition of the Michaelis-Menten constant? Even definition was given in the exam, sir. It is nothing but concentration of substrate only, sir. Michaelis-Menten constant Km is nothing but concentration substrate only, concentration of substrate only, but when at half the maximum velocity. Now in this diagram you're able to see the maximum velocity here. Somewhere in the middle there will be half the maximum velocity. At half the maximum velocity, yes, whatever is the substrate concentration here, that will be the Km. So what is Km, my dear friends? Nothing but the substrate concentration at half the maximum velocity, Now what about this Line-Weaverberg graph? Line-Weaverberg graph is actually the double reciprocal graph. That means, yes, whatever the substrate concentration that we have taken on x-axis, it will be 1 by substrate concentration. And whatever velocity that we have taken in the y-axis, it is 1 by velocity here. It is double reciprocal, and this time it is actually a straight line. The graph will be like a straight line here, which is intercepting the y-axis. Now wherever this line is touching the x-axis, at x-axis you'll be getting the Km, and here it will be 1 by Km. And wherever this graph is touching the y-axis over here, on the y-axis you'll be getting the Vmax, and here it is 1 by Vmax. So it is like double reciprocal graph, my dear friends. Double reciprocal graph. Now, once you have understood these two graphs over here, that is Michaelis-Menten graph and Line-Weaverberg graph, both of them are giving the correlation between substrate and velocity. A Line-Weaverberg graph is actually the double reciprocal one, fine. But how they are actually giving in the exam? In exam, they will be giving you the graph something like this here. With the inhibitor. So enzyme inhibition and the two competitive and non-competitive inhibition, very much important. यहाँ से आपके January exam में एक सवाल पक्का आएगा. Definitely one question will come in your exam, my dear friends. Okay, now first in the exam when this graph is given, try to identify. This black colored one here is actually normal graph. Normal, that means without the inhibitor. And now you are able to see the red colored one which is having competitive inhibitor. Sir, how can you tell this one is the competitive inhibition only? How can you tell it is competitive inhibition? You can very clearly see here, normally without the inhibitor and with the inhibitor, it is meeting at the same place. If it is meeting at the same place, that means the Vmax value is the same. The Vmax will remain the same, but whereas the Km value will actually increase. Vmax same, Km increases, that means it is competitive inhibition. Or that one competitive. Now compare with the normal one with this blue colored one, non-competitive one. So you can clearly see the Vmax will be over here, but for this one the Vmax is here. So therefore what is happening here? The Vmax value is actually decreasing. If the Vmax decreases, yes, the Km is going to remain the same there, and there, yes, my dear friend, that one will be the non-competitive inhibition. And that is what exactly I want you to remember for your exam. Welcome here, different types of inhibition here. So just now I told you about the competitive inhibition. In competitive inhibition, Vmax will be remaining same and the Km will increase. ओके जब कभी भी कंपटीशन होता है वेलोसिटी सेम रखने के लिए वी मैक्स सेम रखने के लिए काम ज्यादा करना पड़ता है काम ज्यादा करना पड़ता है रिमेंबर लाइक दिस ओके ना सो इन कॉम्पिटिटिव इनिबिशन वेलोसिटी विल बी द सेम वी मैक्स विल बी द सेम वेयर एज द Km increases fine. In the non-competitive inhibition, what is going to happen? Exactly opposite of that one, Km will remain the same, whereas Vmax increases. First, you remember these two perfectly. 110% you should be perfect with the competitive and non-competitive. If you are perfect, then even look for the remaining two, sir. Yes. What is happening in the uncompetitive inhibition? You can see both the graphs are going parallelly there. Both the graphs, if they are going parallelly, yes, Km also decreasing, Vmax also decreasing here. Fine. Now similarly, my dear friend, mixed inhibition में क्या होता है? नाम से ही हमको समझ में आ रहा है. From the name itself, you are able to understand. It's like kind of opposite, sir. Km increases, but whereas Vmax decreases. It is like mixed one. But for your MCA exam, I'm telling you again and again that competitive and non-competitive, these two are very much important. If you are perfect with that one, and after that, if you still have time and energy to learn, please go for the uncompetitive and the mixed inhibition because that is not normally asked in your FMG exams. ये दोनों बहुत इंपॉर्टेंट है. क्लियर? चलिए आगे बढ़ते हैं. Iso enzymes. Iso same enzyme catalyzing the same reaction but in different tissues. One of the iso enzyme will be LDH. And LDH has got how many, sir? Lactate dehydrogenase has got 5 isoenzyme. Thank God the name is very simple. LDH 1, 2, 3, 4 and 5. LDH 1 and 2 will be present in the heart, whereas LDH 4 and 5 is in the skeletal muscle. LDH 3 will be in the multiple places. Fine. But here, why I'm telling you about this one here? Yes, my dear friend, correlate our biochemistry with pathology and medicine. LDH 1 and 2 is present in the heart. And that is why during MI, what is going to happen? Yes, LDH flipping will occur. LDH flipping hota hai MI mein. One more isoenzyme will be the creatinine kinase here, and that will be having 3 isoenzyme CKMM, CKBB, CKMB. CKBB will be present in brain, and CKMM will be present in skeletal muscle, and CKMB will be present in the heart. And again the same thing. I hope आपने MI पढ़ा होगा, myocardial infarction. In myocardial infarction, yes, even CKMB is also one of the marker of your MI. Marker of MI. So these two topics you have to revise in enzyme topics, sir, before going to the exam. Is it okay? So very much important about the inhibitors as well as the isoenzymes. Now, my dear friend, welcome to the jackpot in biochemistry. Okay, vitamins topic. Okay, and why I'm telling you the jackpot here, you can expect in your January exam 4 to 5 questions from here, sir. 4 to 5 questions will be coming always from vitamins topic. And always our students are like overconfident. vitamins तो हमने already पढ़ रखा है स्कूल से, बचपन से पढ़ रखा है, कौन रिवाइज करेगा, ये तो आता ही है ना. ओवर कॉन्फिडेंस नहीं चाहिए, कॉन्फिडेंस चाहिए. I request all of you with the folded hands, please revise this topic of vitamins while going on the way to the examination hall. Okay, I hope like many of the students will be having my LMR last minute revision hai, 5 pages wala, 5 pages. In that everything is written over there. Biochemistry has been concise there, and that LMR PDF you can get it anywhere. You know the ways how to get it. So in that PDF, only 5 pages, my dear friend. Please take a printout of that one and keep it ready with you. And my dear doctors, प्लीज रिवाइज दैट वाइल गोइंग टू द एग्जाम ऑन द वे. जब आप मॉर्निंग ऑन द वे आप जाते रहते हो ना, तो तब आप उसको रिवाइज करके जाना, बिकॉज 4 to 5 marks matters a lot. And in the last July exam, my dear friend, remember, they have given exactly 5 questions from vitamins. Only vitamins topic they have given like 5 questions from there. ठीक है ना? Clear with this one here? So vitamin topic is very much important, my dear friends. Fine. Now let's get started with the first one here. Vitamin A deficiency. Can you just give me a minute? I'll be back in a minute. I'll get some water. Yes, my dear friends, I'm back here. Vitamin A deficiency. Now, everyone 200% alert here, my dear friends. Vitamin A deficiency leads to what? Yes, of course, it will be leading to xerophthalmia. Now, let us discuss everything about the vitamin A that is required here, sir. Fine. Given picture depicts the deficiency of which vitamin here? This question was given in FMGE June 2022 here, my dear friends. You can clearly see that there is a white foamy structure over here on the sclera. Yes, that will be due to, that is known as what? Bitot spot here. And bitot spots will be visible when there is a vitamin A deficiency, sir. Vitamin A will be deficiency. Fine. So these two questions has been already asked in your exam. Now let us discuss everything about the vitamin A here. Now, first of all, whenever there is a vitamin A deficiency, I hope you know you know about this classification of xerothalmia over here. Now, XN actually stands for the necrolopia stage, then X1A conjunctival xerosis here, and then X1B will be the bitot spot, X2 will be actually the corneal xerosis, X3A will be the corneal ulcers but involving less than one third of the cornea, X3B will be the corneal ulcers again but involving more than one third of the cornea, more than one third of the cornea. I hope you know this staging over here. Now, after that one, yes, what are the different forms of vitamin A here, sir? Vitamin A has got like three important forms here. Number one, there will be retinol, then there will be retinal, then there will be retinoic acid. From the name itself, we can understand here, retinol, it will be having alcohol functional group, retinal will have aldehyde functional group, and retinoic acid will be having the carboxylic acid functional group, COOH. Okay, now out of this, I'll be covering like almost like many questions over here, sir. Please listen very carefully here. Retinol. Retinol is the one which is absorbed form as well as transported form and as well as the storage form of the vitamin A, already asked in your FMG exams. And then retinal, that is actually present in the rods and cone cells of the retina, that is in the form of rhodopsin. और rhodopsin अगर आपको पता होगा, it will consist of 11 cis retinal here, sir. Okay, along with opsin. 11 cis retinal along with opsin there. Retinal form will be there, aldehyde form. And retinoic acid, you know very well, ATRA, ATRA, all trans retinoic acid, as well as the 13 cis retinoic acid, sir. 13 cis retinoic acid. Fine. And then all trans retinoic acid here, ATRA, this is required for growth and differentiation of epithelium, whereas 13 cis retinoic acid is required for decreasing the keratinization of the epithelium, decreasing the keratinization of epithelium. That is the reason why, my dear friends, remember, that is the reason why, my dear friends, please remember that, yes, if this retinoic acid is deficient, then what is going to happen? It cannot decrease the keratinization. So therefore the keratinization will increase there in the sclera, जो मैंने आपको अभी बताया था पाइटॉट स्पॉट वाला, है ना? तो इसीलिए ये बहुत इंपॉर्टेंट है. और अगर यहाँ पर ग्रोथ और डिफरेंशिएशन ऑफ एपिथेलियम नहीं होता है तो ऑब्वियसली द एपिथेलियम प्रेजेंट ऑन द surface of our skin here is not growing and differentiating there. So therefore, my dear friends, remember what is going to happen. The skin will be like dry, rough and scaly. Dry, rough and scaly skin will be there. So these are all the points that you have to revise for your vitamin A topic here. So next one. A person has a history of alcoholism for 20 years Actually, I've written few things over there. I told you in the beginning, so that we don't waste our time in writing there. So please try to be like very much what do you say, fair enough with yourself. Don't look at the answer there. Before looking at the answer, you try to read the question and try to understand. A person has a history of alcoholism for 20 years. He avoids alcohol last 2 days. Present with agitation, global confusion and disorientation, hallucination, fever, high BP, diaphoresis, and then autonomic hyperactivity. The most common cause of the given condition. Now the problem here is that the moment students are looking at this global confusion word in the exam, khatam, the only thing which is coming into their mind that they have learned in the psychiatry that Goa, Wernicke-Korsakoff syndrome, Wernicke encephalopathy. हाँ, many of them will think global confusion बोलते हैं ना, वही सोचते हैं ना. Please remember here, in Wernicke encephalopathy, what is going to happen? In Wernicke encephalopathy क्या होता है? गोवा होता है. Fine. In this Goa, there is global confusion. Okay, I agree with you. It is given in the question. O stands for ophthalmoplegia. Ophthalmoplegia. Is it given there in the question? Not given. 8 stands for ataxia. Ataxia क्या सवाल में दिया गया है? नहीं. तो there is not a combination of global confusion, ophthalmoplegia and ataxia. ये नहीं है. तो obviously it can't be Wernicke encephalopathy. It is not Wernicke encephalopathy. Then what about the Korsakoff syndrome? In Korsakoff syndrome, what is going to happen? There will be amnesia. Amnesia, है ना? There will be amnesia. Loss of memory will occur. And along with that, there will be confabulations will occur. ओके, एमनीशिया होता है, कन्फैबुलेशन होता है, एज वेल एज दिल बी लाइक सेंसरी एग्नोसिया भी होता है. ओके ना, सेंसरी एग्नोसिया भी हम कहते हैं. ठीक है, फाइन. सेंसरी एग्नोसिया. Now, this is not given here in the question here, so it cannot be the Korsakoff syndrome here, sir. Okay, तो ये तो possible ही नहीं है. Now, the thing where the students are getting confused is about the global confusion. ये कहाँ पर होता है? Please remember in the alcohol withdrawal. In alcohol withdrawal, it is actually known as what, sir? Delirium tremens कहते हैं. Which will be 3rd to 10th day of the alcohol withdrawal. आपके यहाँ पर क्वेश्चन में क्लियरली दिया गया है. Yes, the alcohol has been withdrawn just 2 days back. So 3rd to 10th day of the withdrawal, you'll be able to see delirium tremens. Now, what is going to happen here? यहाँ पर भी global confusion होता है, but along with hallucination and nightmares. Please remember that that combination very clearly. Global confusion, hallucination, along with the nightmares, and along with this, we will also find the physical signs like increased heart rate, increased BP. Yes, of course, tremors will be there. Name itself is given as delirium tremens there, as well as fever will occur. So high fever, high BP, and all these things are given here. So are you understanding now? So therefore, the correct answer for this one, please don't look at the global confusion and mark the answer there as Wernicke-Korsakoff syndrome. No, it is actually the alcohol withdrawal, delirium tremens. Okay, और ये किस में होता है सर? Wernicke and Korsakoff syndrome, that will be due to the vitamin B1 deficiency, thiamine deficiency. So in this manner, my dear friends, remember, yes, in one question itself, we are actually, done with the vitamin B1 deficiency also. Next concept. Which of the following vitamin is deficient in the depicted picture given below here? It was asked in FMGE June 2022. In the picture, you can clearly see, yes, there is stomatitis. Stomatitis, okay, now, angular, stomatitis over here. Nothing but the chelosis, and this will occur due to the deficiency of which vitamin, my dear friends? Remember, vitamin B2, and B2 will be the riboflavin. So vitamin B2 deficiency will lead to what, guys? Yes, angular stomatitis, the chelosis one. That is very much important here, sir. Okay, the next one. अगर vitamin B2 का deficiency होता है, if the vitamin B2 is deficient, then how you are going to estimate this one? This was given in FMG June 2021. So which vitamins deficiency will be there, and they are estimated by which enzyme activity? Let us look at all of them here one by one. If there is vitamin B1 deficiency, sir, that is thiamine deficiency, that is estimated by the activity of RBC transketolase enzyme. Important one. If there is a vitamin B2 deficiency, that is riboflavin deficiency, that is estimated by RBC glutathione reductase activity. Similarly, if there is a vitamin B6 deficiency, pyridoxine deficiency, that is estimated by xanthurenic acid in urine, but after the tryptophan load. So you have to remember these three things here, which vitamin will be deficient, and we are going to see the activity of which enzyme there. So coming back to here, agar vitamin B2 deficient hoga, तो obviously we have to look for the activity of glutathione reductase in the RBC. Clear? Now, once you are done with this one here, a person presents with diarrhea, dermatitis, and dementia. How can you ignore this question? How can you leave this concept and go to the exams? है ना? Diarrhea, dermatitis, and dementia. Every exam, like one question for sure. Okay. What is the probable cause of this one? Yes, it is actually pellagra, the 3D disease. And if you don't treat this one, अगर आप इसको treat नहीं करोगे, तो fourth D होगा, जिसको हम कहते हैं death. So diarrhea, dermatitis, and dementia. Yes, if it is not treated, then it will lead to what, guys? Death. Fine here, which happens in pellagra. And in pellagra, yes, one more very important thing, the image-based question जो हमारे exams में दिया जाता है, जो dermatitis होता है, that dermatitis will be actually the photosensitive dermatitis. In photosensitive dermatitis means only those parts of the skin will be getting, undergoing inflammation which are exposed to light. And a photosensitive dermatitis. That is why there is a necklace which is formed here, and this necklace will be referred to as the Cazel's necklace here. Cazel's necklace. And yes, of course, it is due to deficiency of which vitamin here? That is your vitamin B3, niacin. Cazel's necklace, fine. Now I have given you certain more pictures also regarding this one. Yes, whatever possible images were there regarding the Cazel's necklace I have given you, because in every exam they're giving some or the other image, guys, either this one here or this one here. Yes, so I've given you all the combination of the pictures regarding the Cazel's necklace. Yes, it is seen in photosensitive dermatitis that is happening in the pellagra. Pellagra is due to deficiency of vitamin B3, niacin. Next one. Given picture depicts the deficiency of, you can clearly see in this picture, it's actually the congestion is seen around this one here, my dear friends, cornea. That is circumcorneal congestion. And circumcorneal congestion is also happening in the, yes, deficiency of the vitamin B3. Vitamin B3. That is why I've included this one. Don't get confused here, sir. Fine. A woman with the complaints of pain, tingling sensation, and numbness of the foot has a history of taking vitamin supplements. Taking vitamin supplements. This time it is not deficiency, it's actually toxicity here. Which of the following is responsible for this patient condition? It is actually due to the increased intake of vitamin B6, pyridoxine. Okay, so when you have like more intake of vitamin B6, it will lead to what, guys? Toxicity of that will lead to, yes, the tingling sensation, numbness, and pain in the foot and all. Next one, a micronutrient that is deficient following the post-ileal resection is terminal ileum is responsible for absorption of vitamin B12. If you have done the ileal resection, then what is going to happen? Yes, absorption of vitamin B12 will be compromised. Okay, now if vitamin B12 is actually not absorbed there, then what is going to happen with deficiency of vitamin B12? It will lead to megaloblastic anemia. So one more question which was given in June 2021 in the same manner. A vegetarian patient presents with anemia and hypersegmented neutrophil as shown below. You can clearly see hypersegmented neutrophil. The most probable cause of this one, of course, it is seen in the megaloblastic anemia here. So are you understanding, my dear friends, all the concepts here? So everything is like very well planned here. I have kept all the concepts one by one. So we are seeing all the vitamins like B1, B2, then now we are seeing the B3, B6, B12 one by one, sir. Now, megaloblastic anemia will occur due to deficiency of vitamin B12, or it will be due to deficiency of what's the folic acid, vitamin B9. Megaloblastic anemia will occur due to deficiency of vitamin B12 or vitamin B9. How to do the differential diagnosis? In both of them, what is going to happen? Homocysteine levels will increase. But in vitamin B12 deficiency, additionally, even the methyl malonic acid is also going to increase. Okay, so if you are able to see in the question, this was given in your FMGE exam as a clinical question, my dear friend. So in between the lines, if they give you only homocysteine elevated, it is folic acid deficiency. Suppose if homocysteine and methylmalonyl CoA, both of them are elevated, then it is vitamin B12 deficiency. We have learned about this in detail in our regular classes. I told you how it will happen, all the reactions and everything. But for now, in our last minute revision session of biochemistry, my dear friend, this one, please remember that one word can save 1 mark for you. Is it okay? चलिए सर आगे बढ़ते हैं. Now, deficiency causing the tingling sensation. Now, why I have given you this question here is the moment you see this tingling sensation, obviously you'll be thinking about the peripheral neuropathy. Peripheral neuropathy, you'll be thinking about vitamin B12, है ना, vitamin B12 वाला. अब यहाँ पर options में अगर आप देखोगे, vitamin B12 is not given in the option here. Anywhere you don't find vitamin B12. Then if it is not given, then the better option will be your vitamin B1. Vitamin B1 deficiency will also lead to what, sir? The tingling sensations and all those things. That's all. So these are all like concepts we have, revised about all this one, my dear friends, all the vitamins. In this, I did not discuss about the vitamin C. Vitamin C is going to come ahead. We are going to discuss about the collagen wala part, post-translational modification of collagen. Is it okay? I think it will take another hardly another 5 or 10 minutes of time, my dear friends. And just be along with me. We'll complete this. And then your entire biochemistry part will be done, sir. Welcome to hemoglobin topic. Always again the same thing. One question is coming from here. You might be thinking, what, sir? From this topic, how they are asking the question? Let me show you. Degradation of heme. So whenever your RBC is undergoing lysis, whenever the RBC is going to break down, the hemoglobin present inside the RBC will be released outside. Hemoglobin will have two parts here. So one will be the heme part, another one is the globin part. We know that globin will be the protein part. Okay, and if it is a protein part, it is going to break down to form amino acids, and amino acids will be reutilized in our body. They'll be vapas reutilized in our body. They have got many important functions. Now, similarly, what is going to happen with the heme part? Heme is the non-protein part that will undergo degradation to finally form bilirubin. Now, what is going to happen with the bilirubin, my dear friend? Remember, bilirubin has to be excreted out. But the problem is that it is insoluble. If it is insoluble, my dear friend, what to do? To make it soluble. And to make it soluble, what we're going to do is, we are going to add a substance known as UDP glucuronic acid. When you attach this one, this reaction is known as the conjugation reaction. Conjugation reaction, and then this bilirubin will become bilirubin diglucuronide. And this one here will be the obviously conjugated bilirubin. And this one here will be the unconjugated bilirubin. Unconjugated bilirubin is actually insoluble. हमने क्यों किया conjugation? Why we have done the conjugation? To make it soluble. This is like soluble bilirubin. इतना आसान है. Many of the students will be actually confused with this terminology here, sir. Now, welcome to my class over here. This is like damn easy. Why are we doing the conjugation? To make it soluble, my dear friends. To make it soluble here. Understanding? And once it is soluble, of course, it will be excreted out into the, from the liver, it will be going out into the intestine. And in the intestine, what is going to happen? That urobilin, stercobilin, you know that very well, guys. Okay, now let me do one thing here. Let me just highlight this part here, conjugation wala part here. So I told you very clearly, this bilirubin here will be the unconjugated bilirubin. This one here will be the conjugated bilirubin. Unconjugated bilirubin will be insoluble bilirubin, and this one here will be the soluble bilirubin here. So basically, what is the reaction we are doing here? We are doing the conjugation reaction. Now from here, your examiner is asking you the questions here, sir. Question number 1. Conjugation reaction will increase solubility or decrease solubility? No doubt about that. No need to mug up at all, my dear friend. We are seeing with example over here that yes, conjugation increases solubility. It is increasing the solubility. Number 2, my dear friend. Recently it was asked in your exam. Conjugation belongs to which type of detoxification reaction? Phase 1 or phase 2 reaction? So please remember, conjugation reaction belongs to phase 2 reactions. It belongs to the phase 2 reactions of detoxification, जिसको हम कहते हैं metabolism of xenobiotics भी कहते हैं. So in metabolism of xenobiotics, there will be like phase 1 reaction and phase 2 reactions. Phase 1 will be including that oxidation, reduction, hydrolysis, but whereas conjugation is phase 2 reaction. Even this is also covered here, sir. So even in our, LMR here, last minute revision here, I'm including even the small points also, my dear friend. So that's the reason why I'm telling you repeatedly. You can see every topic is done here, and that too in a detailed manner. Try to revise this video again and again. It will be surely available till your exams. Don't worry about that one. It will be available on my on my YouTube channel. On my YouTube channel, that is Dr. Azam lectures. So you can find there, and you can actually revise it multiple number of times. I'm not going to remove the video. It will be there till your exam. So we are also doing like small things also. So please revise this one. Now welcome to the minerals topic here, sir. Few questions here and there. Which of the following is not a micro element? Okay. Now first you should understand what is the difference between the micro and macro. Micro, the RDA will be less than 100 milligram per day, and macro if the RDA is more than 100 milligram per day. Micro examples will be, of course, all your vitamins here, as well as the iron, zinc, copper, manganese, molybdenum, and all those things will be the minor. Whereas macro will be the calcium, magnesium, potassium, phosphorus, and all. All this will be the major macro ones. So they are divided based on what? They are divided based on the RDA value. If the requirement is less than 100 milligram per day, that will be like micro. And if it is more than 100 milligram per day, that will be macro. Now, which of the following is not a micro element? Of course, the answer here will be calcium. Remaining all are micro elements. And here, my dear friends, because we are discussing about this one, please remember about the selenium. About selenium also, they are asking certain questions. Number one, selenium, it is required for which enzyme? It is required for number one, glutathione peroxidase enzyme. At least remember this enzyme, my dear friend. There are like another set of another enzymes also. Like selenium is required for other enzymes also. Please remember, like at least this one here, glutathione peroxidase requires selenium as a coenzyme. Selenium is acting like a coenzyme here. Or agar मान लो there will be deficiency of the selenium over here, it will lead to Cushing's disease. Cushing's disease, your previous year exam question already asked in the FMGE, sir. And here in the Cushing's disease, the person will die because of cardiomyopathy here, sir. Okay, and that is due to decreased selenium here. Is it okay? Next one here, my dear friend. Menkes disease is caused due to, see, of course it is due to the copper, but the problem here is that is it due to the copper deficiency or copper toxicity? So please remember in Menkes disease it is due to, yes my dear friends, remember copper deficiency. और आप सबको पता है इसमें क्या होता है? What is happening in this one here? Yes, the kinky hair appearance will be seen in this one here in the Menkes disease. Copper deficiency is important here. Next one, yes, they are giving the questions regarding the marasmus and the Kwashiorkor also. Now, what are the differences between the marasmus and Kwashiorkor? In marasmus, yes, there is a total calorie deficiency. That baby is not at all fed at all, total calorie. But in Kwashiorkor, proteins are deficient there. Okay, age, when will marasmus occur? In the infants, whereas Kwashiorkor will occur in the 6 months to 3 years of age. Appetite, yes, of course, because they are totally not fed, it will be voracious appetite will be there. But here, because the proteins are fed here, there will be lack of appetite. Now, because of the proteins, what is going to happen? I hope you have learned in physiology the changes in the oncotic pressure and all those things. That is the reason why the fluid will be accumulated, yes, leading to edema in the Kwashiorkor, as well as यहाँ पर ना fatty liver भी होता है. This will also occur here in the Kwashiorkor one, sir. So in this manner, my dear friends, we are able to yes differentiate here between the marasmus and the Kwashiorkor. Let us see the questions that are given in the exam here. Fat, sugar, baby is seen in marasmus or Kwashiorkor? Is of course in the Kwashiorkor. I told you just now, edema will occur. Fat, sugar, baby will be seen in Kwashiorkor. And after that, the boy presents with the voracious appetite. Voracious appetite will occur in the marasmus here. And then along with that, there is overall swelling also. Oh my God. There is voracious appetite will be there, that is marasmus. Overall swelling edema, which will occur in Kwashiorkor. The answer here is marasmic Kwashiorkor. That is marasmic Kwashiorkor will occur. And this question was given in FMG January 2022, my dear friends. Okay. Next one, moving ahead with the last and final thing right now, that is your molecular biology. In molecular biology, I'll tell you certain, things which are important for your exams. Number one, nucleosome. They're asking the questions regarding the nucleosomes, so the arrangement of the structures in the nucleosome. In the center of the nucleosome, there will be, yes, octamer of histone proteins. There will be like 8 histone protein. There will be 2 molecules of H2A, H2B, H3 and H4, sir. So in the center, you'll be having octamer of histone protein. And that will be coiled by DNA. So DNA plus histone is nothing but nucleosome. Acha, if you look at the names here, I started with H2A, H2B, H3 and H4. Where is H1 then? So remember, H1 will be present along with the linker DNA, which is going to link the 2 nucleosomes. Fine. DNA has got the negative charge, of course, because of which molecule? That is because of the phosphate group, that is PO4 minus 3 group here. But histones are having positive charge, that is because of lysine and arginine. Because of more of lysine and arginine amino acid, it has got the positive charge. These are all like MCQs, important MCQs asked in your exams. Please remember all the points. One point is also important here. Next one. In liver, ApoB gene is completely translated to ApoB100, whereas in the intestine it is translated only to ApoB48. So there is ApoB gene and that is going to become ApoB100 in the liver. Okay, but in the intestine, what is going to happen? Only it is ApoB48. I hope you remember when we studied about the chylomicron and all. Which of the following mechanism explain this one? Please remember it is due to what? RNA editing here. Now please look at this diagram here, my dear friends. From we know that from the DNA we are going to get the RNA here. This is by transcription. यहाँ तक कोई दिक्कत नहीं है. No problem till here. Okay. Now what is going to happen after the RNA? In the liver, what is going to happen? Yes, it is not at all edited. It is perfect there. So if it is perfect, what is going to happen? All 100% of all the amino acids will be attached there. That is the reason why it is known as ApoB100 there. But in the intestines, what is going to happen? That RNA will be edited there. And because of that, what is going to happen? Before itself, only when only 48% of the amino acids are attached there, at that time itself, stop codon will come there. Because of the editing there, what is going to happen? The stop codon is going to come before itself, like almost just at the 48% of the amino acids will be attached. इसीलिए तो उसका नाम क्या रखा गया है? ApoB48 रखा गया है. Okay, so therefore in intestine it is going to become the same gene only, the same gene, ApoB gene. In the intestine it is going to form ApoB48, but whereas in the liver it is going to form apoprotein B100, जो VLDL में होता है, VLDL. Okay, और ये वाला जो होता है ApoB48, it will be present in the chylomicrons. Okay, so what is the reason for this one? Yes, the reason is at this level, what is happening here? RNA editing is taking place. So therefore, what is the answer for this question here, sir? RNA editing here. Next one. Now, once you are done with this one, tRNA structure was asked in your FMGE exam. So please look at this structure before going to the exam once. We all know it is like clover leaf shaped structure. You have to remember that one of the end will be 5', another end will be 3'. At 3' end, we are going to attach the amino acid. That is why it is known as acceptor. Exactly opposite of that one is the one which is going to read the codon here. So because it is going to read the codon here, it is anti-codon arms. Please remember that one. In the image-based question, they have asked about the anti-codon arm. And the remaining two arms, just read it once and leave it. They will never ask you. One is your DHU arm, dihydrouridine arm. Another one will be the T-Cy-C arm over here. And then there will be a variable arm is also present over there. That is the structure of tRNA, transfer RNA, my dear friend. Fine. Next one, please read this question here. A 22-year-old male returning from China was found to have fever. Of course, we are actually, the examiner is actually indicating about what? The COVID wala part. What is a specific diagnostic test that needs to be performed? अगर आपको याद होगा वो COVID वाला era जो था 2020 and 2021, yes, every time we were doing that RT-PCR, reverse transcriptase PCR, reverse transcriptase PCR. Of course, why? Because DNA to RNA will be actually what here, guys? That is nothing but transcription. But here, tell me, the COVID virus, it was actually the RNA virus. तो पहले उसको क्या करना पड़ेगा? वापस DNA में convert करना पड़ेगा. That is why it is reverse transcriptase PCR. So what is the specific diagnostic test here? That is the RT-PCR. Perfectly done. Next one. Which of the following plays a role in the collagen modification? Yes, my dear friend. Nowadays, from this topic, the questions are being asked in your exam. Now, I'll just tell you the important thing here. In collagen modification, what is going to happen? Yes, hydroxylation will take place. What is the meaning of hydroxylation? Addition of OH group. But where are you going to add this OH group? Please remember at lysine and proline. Sir, what is the meaning of this one? The meaning of this one here is collagen is a protein. Protein means it is made up of amino acid here, polymer of amino acid. It is made up of many amino acid. Somewhere in the middle, we are actually having lysine. Okay. Then again, many amino acid. Somewhere in the middle, मान लो कि यहाँ पर proline होता है. Is it okay? This is just like our schematic diagram to understand. Now, wherever you have lysine, wherever you have proline, there what is going to happen? OH group will be added here, sir. Hydroxylation at lysine and proline. Are you understanding the meaning right now? Hydroxylation at lysine and proline. Now, what is the name of the enzyme? The name of the enzyme will be hydroxylase. Lysyl hydroxylase. And what is the name of the enzyme here? That is again hydroxylase. And here it will be the prolyl hydroxylase. Prolyl hydroxylase. Okay. Now these two enzyme requires vitamin C as a coenzyme. Okay, that is the reason why vitamin C is very much important. It is required for modification of the collagen. Modification of the collagen, vitamin C is required here, sir. तो तभी तो क्या होता है, that is the reason why I did not discuss with the vitamins topic, sir. Now we are able to discuss perfectly here. So vitamin C is required for which enzyme? Lysyl hydroxylase and prolyl hydroxylase. Is it okay? Then next, number two, my dear friends, please remember that अगर विटामिन सी का डेफिशिएंसी हो जाएगा तो कोलेजन मॉडिफाई नहीं होगा. इफ कोलेजन इज नॉट गेटिंग मॉडिफाइड ओवर देयर, देन व्हाट इज गोइंग टू हैपन? इट इज रिक्वायर्ड फॉर मेंटेनिंग हेल्दी gums. The gums will crack and the bleeding will occur. Bleeding gums that will be seeing in the scurvy. Scurvy में आप जो देखते हो, है ना? Next one, sir. Apart from this, one more important thing that you have to remember. Yes, in this manner you have to form like three chains here, and those three chains add the OH group. Wherever you have the lysine and proline, you add the OH group, and then what is going to happen? They are going to form a triple helix. Last July exam question, triple helix formation. Triple helix formation आपके last July exam में पूछा गया था. Okay ना? And one more point you have to remember is that there is an enzyme known as lysyl oxidase. And lysyl oxidase, why it is important for your exam? Why? Because this enzyme requires copper as a coenzyme. Copper as a coenzyme. और अभी भी तो मैंने आपको बताया है, deficiency of the copper, what is going to happen? Collagen will not modify properly. The hairs will become like kinky. Kinky hairs will be there in that one. मेन किस डिसीज में हमने देखा था, ठीक है ना? So therefore, my dear friends, welcome back here. In collagen maturation or modification, what is required? Copper is required along with vitamin C, that is ascorbic acid. Copper is required along with the vitamin C there, guys. Okay. And by this, my dear friends, these are all the important topics that you have to actually revise for biochemistry. Is it okay? So if you people have followed my entire lecture, it's exactly like 2.5 hours here. In 2.5 hours, we have completed the entire biochemistry, electron transport chain, carbohydrate metabolism, protein, lipid, nucleotide metabolism, and then even about the molecular biology, whatever required topics were there, enzymes, hemoglobin, everything has been done in detail, my dear friends. Very neat and clearly I have explained you over here. Repeatedly, I'm telling you again and again, yes, my dear friends, revise these topics, revise this video at 2x speed, how many ever times as possible. Keep a reminder in your phone, and every now and then you try to revise that one. Don't forget to revise before going to the exams. Okay, and on the screen over here, you can see the QR codes of the YouTube channel, Dr. Azam lecture over there, and then my Instagram handle, Dr. Azam Anatomy, as well as the Telegram channel there. So please, like, share, and subscribe over there, and please share with as many friends as possible so that everyone will get benefited. Because please remember one thing that FMG exam is an exam where it is just a screening exam. Everyone has to clear. It's not like competition to get like good rank and bad rank. So please help everyone. Believe me, whatever God you follow, there is some energy that is going to always help you if you help others. अगर आप दूसरों को हेल्प करोगे तो ऑब्वियसली कहीं ना कहीं से आपको ना, जैसे हम कहते हैं, जहां पर आपका गुमान भी नहीं होता है, जहां पर आपकी सोच भी नहीं होती है, वहां से आपको मदद मिलेगी एग्जाम में. तो हेल्प एज मेनी पीपल एज पॉसिबल, माय डियर फ्रेंड्स. डोंट बी इन अ कनिंग मैनर कि आई एम नॉट गोइंग टू हेल्प इन ऑल दिस. ठीक है ना? सो लाइक, शेयर एंड सब्सक्राइब दिस वन. प्लीज इन्फॉर्म एवरीवन अबाउट माय यूट्यूब चैनल टू एवरीवन. आई एम गोइंग टू कम अप विद many videos on that one. Apart from that, on my Instagram channel, I have started posting like reels of the important, very high yield topics of anatomy also. You try to revise all those reels and go to the exams. Is it okay? So by this, my dear friends, that's all from me here. Thank you all for listening to me very patiently. Thank you all, my dear friends, and hope you all come out from your FMG exam with flying colors. And yes, of course, God bless you all. God bless you, my dear friends. Bye bye. Take care. Be happy. Stay healthy.